benefitnya apa setelah dilakukan modifikasi tadi, tapi ada tantangan ya atau batasan. Misalnya, mengecilkan ukuran partikel itu gampang-gampang susah. Sudah kecil, ya kan energi bebas permukaannya besar. Jadi dia cenderung untuk stabil dengan cara agregasi. Jadi mengecil sudah dikecilkan membesar lebih besar dari awalnya bisa jadi ya. Alirannya menjadi kurang baik, aliran serbuknya. Ya, jadi itu harus diantisipasi ya. Untuk pembentukan garam mungkin tidak kan ini senyawa kimia ya, berarti bisa jadi tidak stabil ya. Stabilannya berubah atau dia sangat tergantung dengan pH ya. Dan sebagainya lah ya. Amortisasi itu bentuk tidak stabil loh. Ya karena dia tidak teratur. Ya, kisi-kisi kristalnya tidak teratur, jadi dia cenderung teratur. Nah, bahan baku obat aja senangnya teratur gitu ya. Kita juga harus teratur juga ya. Nah, begitu dia tidak teratur, dia nggak nyaman. Teratur lah. Nah, teratur itu artinya apa? Rekristalisasi. Jadi amortisasi itu tidak stabil. Kalau kita nggak menambahkan stabilizer, mungkin sejalan dengan waktu dia akan kembali ke bentuk kristalnya semula. Jadi kelarutannya berubah lagi. Makanya jangan kaget ya industri kalau sudah disolusinya bagus di awal-awal, setelah 6 bulan kok disolusinya menurun. Banyak yang bingung, kenapa ini? Padahal kan awalnya bagus. Ya bisa banyak hal yang terjadi. Bisa jadi agregasi di dalam produk itu tadi bisa ya perubahan kristalnya dan sebagainya sehingga yang tadinya bagus menjadi kembali tidak bagus lagi. Dan banyak sekali nanti mungkin bisa dilihat kembali ya, ini semuanya materi nanti akan disampaikan kepada peserta. Kemudian nah ini yang modern ya, beda dengan yang modern, pendekatan inovasi ini kita lebih banyak bermain-main dengan material ya. Jadi kita harus tahu tentang ilmu material ya. Kita harus mendalami material science. Ya, bisa jadi nanti ngambil kuliah-kuliah tambahan atau kursus-kursus ya tentang material science, engineering-nya ya, tekniknya gimana, alatnya apa gitu ya. Kemudian ya tadi ya seberapa signifikan solusinya itu bisa ditingkatkan. Oke, ini beberapa strategi ya nanoteknologi yang sekarang sedang marak ya di Indonesia, baik itu pengembangan nanonisasi, nanopartikel, pembentukan nanokristal atau nanoemosi ya, masing-masing punya kelebihan dan kekurangan ya. Tekniknya juga berbeda-beda ya. Kalau nanokristal itu mekanik ya, nanopartikel mekanik top down, jadi memberikan energi yang luar biasa pada partikel besar menjadi partikel-partikel kecil ya dengan menggunakan berbagai metode ya, bisa milling, baik itu wet maupun dry milling ya, kemudian high pressure homogenization, kemudian bottom up ya, anti solvent precipitation ya dan sebagainya. Nah ini saya juga punya tabelnya ya, jadi ini mungkin masing-masing pendekatannya. Jadi lipid based drug delivery ini juga banyak dilakukan, terutama untuk obat-obat lipofilik ya, larut baik dalam lipid. Nah ini teremulsifikasi ya, diemulsifikasikan. Kalau dia diemulsifikasikan berarti di sini ada komponen minyak atau lipid. Nah nanti mekanisme absorpsinya itu beda dengan yang non lipid. Jadi absorpsi itu bisa langsung masuk ke sistem sirkulasi sistemik yang pas metabolisme dulu melalui vena porta hepatica, ada yang melalui limfatik. Ya, limfatik itu untuk yang minyak-minyak. Nah, yang melalui limfatik ini dia menghindari fosfas metabolisme. Jadi beda jalur, ya, beda jalur. Jadi ada benefitnya juga ya. Jadi benefitnya bypass hepatic fosfas metabolisme dan meningkatkan solubility serta permeability karena emulsifikasi tadi di dalam saluran pencernaan. Cara yang lain yaitu hot melt extrusion, kemudian spray drying. Ya, spray drying ini menghasilkan fine amorphous powder, jadi serbuk halus non kristalin atau amorf. Ya, di sini banyak porositas ya yang menyerap nanti jadi pintu-pintu masuk media ya kapilaritas sehingga memudahkan obat tadi bisa terlarut dengan cepat gitu. Kemudian cara-cara yang lain, nah ini yang mungkin di luar nalar tapi terjadi ya, 3D printing ya dari produk-produk farmasi. Jadi emerging tool yang maksudnya adalah mengcustomize ya, customize profil pelepasan obat dan bentuk sedia. Ini sudah ada yang di-approve oleh FDA. Jadi Spritam ini nama generiknya levetiracetam, ini FDA approve ya, first FDA approve 3D printed tablet. Bentuknya seperti ini, nanti bisa juga dilihat di internet bentuk obatnya tabletnya seperti apa. Ya, tapi kartunnya seperti itulah ya. Dan ini modified release ya dengan pola disolusi yang terkendali. Jadi kalau kita bandingkan cara-cara tadi, prinsipnya nanokristal pasti ya sama dengan mikronisasi tadi adalah penurunan ukuran partikel. Benefitnya jadi ultra fast dissolution ya karena sangat kecil tuh, jadi dia luas permukaannya besar, tingkat pembasahannya lebih tinggi. Nah, batasannya apa? Tadi ya mudah teragregasi, jadi perlu stabilizer. Nah, perlu stabilizernya ini juga pemilihannya complicated loh. Tidak satu bisa one for all, enggak. Jadi masing-masing punya pasangan. Jadi kalau yang muatannya negatif, stabilizernya apa dan sebagainya. Kan di kuliah juga sudah disampaikan ya, stabilisasi itu ada yang sterik, ada yang elektrostatik, ya elektrik, ada yang elektrosterik ya, jadi kombinasi. Lalu banyak hal lah ya, 3D printing misalnya, layer by layer fabrication, ini untuk personalize ya, memang untuk personalize, programmable release. Nah, limitasinya adalah masih isu regulasi ya. Tadi FDA sudah approve ya, tapi kan untuk bisa di-approve itu kan harus punya data-data yang sangat kompleks ya. Dan scale up-nya juga mungkin bermasalah. Ini kalau misalnya mau buat 1 juta tablet gimana? Bayangkan aja seperti kita ngeprint. Tapi ternyata waktu saya lihat ya di video itu, yang namanya 3D printingnya itu ya kayak mesin tablet yang dari sini sampai sana. Jadi jangan dianggap seperti printer yang kita punya di meja. Ya, tapi ya itu tadi sistemnya itu ya mesin gitu loh. Cuman dia printing gitu. Jadi dia membuat formula tadi berdasarkan yang kita masukkan gitu. Kita input apa, misalnya laktosa sekian, ini sekian. Ya, nanti pas waktu dijalankan sistemnya sudah ada laktosa dan sebagainya, ya dia tinggal mencampur. Gitu. Jadi jangan dibayangkan seperti printer ya. Tapi ini mungkin istilah ya 3D printing itu seperti di print, tapi bukan seperti printer yang kita punya untuk ngeprint dokumen gitu ya. Kemudian ya ini yang terakhir ya, ketiga nih ya, topik ketiga studi kasus. Jadi cara-cara yang tadi sudah saya tampilkan ini sudah diterapkan di produk yang sudah rilis di pasaran gitu ya, yang dan ternyata memang bisa meningkatkan kecepatan disolusinya. Jadi di sini nanti kita akan lihat pre and post technology dissolution improvement. Jadi dibandingkan before and after penerapan teknologi. Kemudian inovasi formulasi dan peningkatan bioavailability secara in vivo. Nah, ini kita lihat ya tadi ya, nanokristal teknologi. Itu sudah diterapkan di produk namanya Tricor nanokristal yang isinya adalah fenovibrat. Nah, fenovibrat ini untuk yang konvensional dia itu micronize. Untuk yang inovatif dia nanonize ya, nanokristal. Ukurannya kurang dari 200 nanometer. Klasifikasi kelarutannya sama, bisa S2 juga ya. Jadi karena kelarutannya bermasalah nih ya, jadi permeability-nya enggak bermasalah. Kemudian kecepatan disolusi untuk yang micronize itu 60% dalam 1 jam. Kalau yang nanonize itu lebih dari 85% dalam 10 menit. Jadi bisa di wave itu, kan wave itu syaratnya F2 kurang dari 15 menit sudah mencapai lebih dari 85% kan. Absorpsinya, nah karena dia masih bermasalah kelarutannya, jadi dia sangat dipengaruhi oleh keberadaan makanan. Jadi adanya makanan itu bisa meningkatkan atau menurunkan absorpsinya. Sementara kalau yang nanonize itu dia konsisten gitu. Jadi tidak dipengaruhi oleh adanya makanan ya, sehingga bioavailability-nya juga konsisten. Kemudian pendekatan amorfisasi ini juga sudah diterapkan dengan cara amorfisasinya dengan cara hot melt extrusion. Produknya itu posaconazole ya. Jadi posaconazole ini formula konvensionalnya adalah suspensi. Untuk yang inovasinya adalah tablet dengan menggunakan hot melt extrusion tadi ya. Jadi polimer plus active ingredient dilelehkan bersama-sama, kemudian solidifikasi dan kemudian diekstrusi untuk menghasilkan partikel-partikel granul-granul. Tentunya syaratnya adalah untuk obat-obatan yang tahan panas ya. Jadi kalau terbatasannya untuk obat-obat yang tidak tahan panas tidak bisa dengan cara ini. Jadi harus dipilih-pilih. Kemudian bentuk obatnya kristalin, setelah di melting bersama dengan polimer tadi menjadi amorf. Ya, dispersi padat amorf ya karena dia terdispersi dalam matrik polimer. Sehingga kelarutannya bisa meningkat lebih tinggi dan menyebabkan bioavailability-nya itu dari 8% menjadi lebih dari 50%. Ya, dan ini stabil ya, produk akhirnya stabil karena API-nya stabil dalam panas kan, jadi tidak ada masalah setelah diaplikasikan panas untuk menghasilkan amor tadi. Kemudian pendekatan ketiga SEDDS ya, pada siklosporin itu produknya neural. Impact-nya mengurangi variabilitas ya yang tadi disolusinya itu naik turun jadi konsisten dan ini menjadi faktor kritik pada pasien yang mengalami transplantasi. Nah, obatnya metode dari obatnya ini poorly soluble atau dia lipophilic. Neural juga sama sebenarnya lipophilic juga ya. Tapi di sini formulasinya larutan berbasis minyak. Kalau yang inovatif SEDDS in situ emulsion. Jadi in situ itu emulsinya terjadi di saluran pencernaan. Ya, jadi tidak diemulsikan di luar tapi setelah dikonsumsi dia teremulsifikasi di dalam saluran pencernaan sehingga kecepatan dissolusinya menjadi cepat dan konsisten yang menyebabkan bioavailability-nya juga meningkat dan konsisten serta tidak dipengaruhi oleh adanya makanan ya, di dalam saluran pencernaan. Kemudian yang pendekatan keempat itu spray dried dispersion itu sudah diterapkan pada produk itraconazole namanya Onmel ya. Jadi yang konvensional itu sporanol, setelah dia memodifikasi dengan teknologi yang inovatif jadi Onmel. Nah, ini kita lihat perbedaannya ya. Jadi disolusinya yang sebelum menggunakan spray dried itu tergantung pH dan lambat. Setelah dia di-improve tidak tergantung pH ya, dan lebih cepat. Juga tidak tergantung makanan dan bioavailability-nya bisa lebih dari 80%. Kemudian yang ketiga, selanjutnya adalah 3D printed instant release ya, jadi modified immediate release. Ini tadi sudah saya singgung-singgung. Ini kan the first soalnya ya, the first jadi contohnya baru satu. Nah, ini kita bisa lihat di sini ya. Disintegrasinya kurang dari 10 detik dan untuk special population. Cut sebentar ya, 1 menit sampai adanya beres dulu. Baik ya, saya lanjutkan lagi. Jadi berbagai strategi yang tadi sudah saya sampaikan ini tentunya banyak menghadapi tantangan ya, namun ada peluang juga ya, future opportunity di dalam peningkatan disolusi obat ya. Yang namanya inovasi pasti nggak semuanya mudah ya, pasti ada masalah ya. Jadi masalah itu pasti ada, tapi juga ada solusinya gitu ya. Jadi masalah ini saya bagi 4 ya. Nanti pertama stabilitas ya, concern terhadap stabilitas. Seperti hot melt extrusion itu kan tidak bisa untuk obat-obat yang thermal instable ya, jadi ada keterbatasan. Kemudian untuk amorfisasi ya, amorfisasi juga ternyata dia kan tidak stabil ya, dia akan mengalami rekristalisasi. Jadi pasti ada seperti itu ya. Kemudian yang masalah kedua adalah upscaling pada skala industri. Jadi mungkin membuat nanopartikel skala lab gampang, begitu udah berbicara industri mungkin nggak gampang lagi gitu karena mesinnya gitu ya, energinya, kemudian cost-nya gitu banyak sekali hal-hal yang dipertimbangkan. Nah, dan ternyata memang benar belum ada industri farmasi yang saya lihat punya nanomilling begitu. Ya kalau di ITB ada nanomilling skala pilot, tapi untuk skala industri tidak punya gitu ya. Jadi di industri belum ada gitu. Eh ya jadi banyak hal ya yang terbentur pada upscaling di industrial scale gitu ya. Terus kemudian masalah yang ketiga adalah pertimbangan regulasi ya dan biaya. Ya, ini pastilah ya. Belum tentu kita membentuk nano harus ada justifikasi ilmiah apa buktinya gitu ya. Ya buktinya sebenarnya di solusinya lebih bisa memenuhi syarat, BE-nya memenuhi syarat. Tapi ada lagi justifikasi yang lain. Keamanan. Bagaimana prosedurnya? Keamanannya nanti gimana? Nah, biasalah dan pertanyaannya selalu seperti itu kan. Toxicity-nya gimana, safety-nya gimana, terus tanyakan prosedurnya dan sebagainya. Dan biaya juga pasti menjadi pertimbangan ya kalau sudah sampai ke industri farmasi ya karena kan persaingan harga juga ya. Kemudian masalah yang berikutnya adalah personalized medicine ya untuk yang 3D printing tadi. Ya jadi kan terbatas untuk kalangan tertentu dan juga jika kita mendesain formulasi ini berbasis AI. Ya sekarang kan sudah diinginkan penetrasi AI pada semua sektor ya termasuk sektor industri farmasi. Apakah sudah menerapkan AI dalam pengembangan produknya dalam R&D-nya ya. Jadi eh, di sini ya AI and digital tool ini eh, deskripsinya apa? Jadi machine learning yang digunakan untuk pemilihan excipient. Ya kita sudah belajar ya di perkuliahan supaya tidak mengembangkan formula itu sampai ratusan gitu ya, puluhan bahkan ratusan kan sudah menggunakan modeling ya, factorial design atau apa. Nah, sekarang sudah ada AI dan digital tool ini juga akan membantu membuat itu tadi ya, prediksi formula sehingga lebih efisien, tidak mengganggu tenaga, biaya dan waktu. Ini sangat untuk industri farmasi sangat worth it gitu ya. Juga di dalam memprediksi kinetika disolusi. Ya bahkan bisa memprediksi kinetika disolusi sebelum kita melakukan uji disolusi. Ya kalau uji disolusi misalnya yang UDT itu kan ya, udah banyak pH-nya gitu ya, terus banyak sampelnya juga ya 12 vessel dibandingkan antara komparator dengan uji gitu. Jadi memakan waktu kan. Nah, itu bisa dipangkas dengan bantuan AI gitu ya untuk meramalkan ya nanti dibuktikan. Itu namanya juga ramalan gitu ya. Dan juga modeling stabilitas. Jadi untuk modeling stabilitas kira-kira nanti pola degradasinya gimana, faktor yang mempengaruhi degradasi dan sebagainya. Nah, ini kalau boleh saya rangkum ya, ini terakhir ya. Jadi challenging-nya itu terkait dengan stabilitas, ya, kemudian skalabilitas, regulatory and cost, dan customization. Ya, jadi di sini kita bisa lihat current challenge-nya ya untuk stabilitas itu kemungkinan terjadi kristalisasi, kemudian agregasi, skalabilitas terkait dengan high cost ya, investasi mesin mungkin ya. Kemudian bisa jadi juga tidak reproducible. Regulatory mungkin ya tadi saya katakan kompleks approval ya juga memakan biaya yang cukup tinggi dan untuk yang 3D printing tadi terbatas pada personalisasi dalam formulasi jadi tidak bisa untuk massal gitu. Tapi ya itu tadi ya ada masalah pasti ada solusi. Ya, solusinya apa ya? Kita dituntut untuk mencari stabilizer yang smart ya bisa mempertahankan tidak terjadi rekristalisasi maupun agregasi ya mungkin dengan cara nano coating dan sebagainya untuk meningkatkan skalabilitas ya dengan menggunakan sistem modular ya atau sistem kontinu dan juga real time analysis gitu ya. Kemudian untuk menghindari regulasi yang kompleks dan biaya yang tinggi ya bisa menggunakan model in vitro in vivo correlation ya kemudian berbasis pada pedoman global harmonisasi itu ya. Dan itu artinya menuntut kita untuk lebih banyak menggali ilmu lagi yang baru-baru tadi ya untuk menjawab tantangan-tantangan yang sebenarnya di balik tantangan itu ada sesuatu yang luar biasa hanya untuk mencari yang luar biasa tadi kita harus smart dalam banyak hal ya terkait dengan masalahnya apa gitu. Saya kira itu saja dari saya, mudah-mudahan bermanfaat dan bisa memberikan tambahan wawasan. Demikian, terima kasih atas perhatiannya. Wassalamualaikum warahmatullahi wabarakatuh.
Now that concludes our morning sessions, and now we will have a break for lunch. And we invite you to enjoy networking with your fellow participants.
Everyone can grab lunch at in front of your chair, and we kindly ask everyone to return to the hall by 1 p.m. for the continuation of the technical sessions. Thank you. they are not so
Ya betul sih, itu sebenarnya udah kayak online yang mana dari lah, ya dia juga itu kan ada arahan sih tidak jalan duluan sesuatu kan katanya kalian juga rasa karena takutnya juga ini kan tentunya konflik jatuh itu kayak gitu kan konflik kan ya sih kalau ada ya
Please guys, and so closely to the walking in my position. It is not 25 minutes. When you start to this, was a obvious 60 over the girl, this is saat saya sudah berada hari ini saya bos sedih, sebenarnya dan cara ini bisa punya sebagai yang mantap, organik sehat sempurna, buat dari 100 dunia yang lada hitam. Dikonsumsi setiap hari, miliki produknya
bersama memasukkan dalam itu dia global sama di teman Islam kayak di termasuk awal itu yang kami dari mana itu sebenarnya jadi
Siapa yang mau membahas sejarah Islam? Mulai dulu, tenang. Kembali ke Rasulullah, kelompok formulasi sebuah cuci yang sertifikasi halal untuk mensucikan tubuh dari najis karena melalui syariat Islam. Lalu, bagaimana cara memakaian? Pertama, bersihkan dahulu bagian yang tercuci. Kedua, buang sabun pada bagian tubuh dan perkenalan najis secukupnya. Lalu rapatkan dan basuh dengan air. Ketiga, basuh kembali dengan air sebentar. Cukup mudah, bukan? Sabun ini juga dilengkapi dengan air. Suplemen herbal yang bermanfaat dan menjaga kesehatan tubuh buat dari fokus kulit dan lada hitam. Dikonsumsi setiap hari. Miliki produknya. Terjadi ini baru sedikit dalam itu agar melakukan tindakan tertentu yang memproses organisasi untuk mengambil yang berjalan tinggi. Istilah yang mudah dimengerti kesehatan hasil penelitian serta bermanajemen terhadap yang adalah komitmen dan terhadap yang dilakukan selanjutnya jika dilakukan dengan ini keseluruhan.
aktif sampai ke menghasilkan efek. Nah, satu hal yang mungkin menjadi kunci dari temuan ini adalah disolusi yang pada banyak kasus, tidak selalu ya, tapi dalam banyak kasus menjadi faktor kunci dari absorpsi suatu obat gitu. Tadi sudah dibahas ya ketika kita memiliki BCS kelas 2 misalnya, disolusi akan ketika kita memiliki BCS kelas 2, maka disolusi menjadi penentu utama dari absorpsi obat. Gitu. Nah, ini mungkin alat-alat disolusi yang ada di lab Bapak Ibu semuanya ya, yang sering kita lihat gitu. Ada dari berbagai merek ya, yang saya tunjukkan di sini gitu, sengaja saya tunjukkan semua merek. Gitu. Nah, ini kan yang mungkin setiap harinya dilakukan di lab-lab Bapak Ibu di industri ya. Disolusi untuk kompendial monograf, disolusi untuk QC, yang tujuannya untuk batch release, itu menjadi suatu hal yang rutin dilakukan. Saya yakin semuanya familiar dengan ini. Kemudian yang kedua, satu tingkat lebih tinggi, disolusi terbanding, yang digunakan untuk membantu uji bioekivalen misalnya ya. Dan juga menjadi satu semacam aturan atau guidance untuk melakukan pengembangan formula juga gitu. Ketika misalnya kurang atau tidak bioekivalen di 1 pH, maka melakukan modifikasi dari sisi formula juga berdasarkan uji disolusi terbanding. Kemudian ini juga yang bisa menjadi satu data yang bisa disubmit untuk registrasi ya, meskipun pada kenyataannya uji BE di manusia tetap dilakukan gitu. Nah, ini kan biasanya sampel-sampel yang kita uji ya, tablet, produk ya, tablet, kapsul gitu. Jadi kalau kita bicara disolusi umumnya ya yang teringat di pikiran kita adalah tablet gitu. Ya, tablet gitu, pasti ngomongin disolusi ya lagi ngomongin tablet gitu. Nah, tapi ada juga uji disolusi lain yang bisa dilakukan, contohnya adalah powder dissolution misalnya, disolusi serbuk. Dan satu yang akan saya bahas di sini, ini kepotong ya. Kepotong slide-nya. Itu adalah disolusi intrinsik gitu. Jadi itu yang akan dibahas pada sesi saya ini. Jadi ini sebenarnya apa gitu ya. Nah, jadi disolusi intrinsik itu adalah disolusi dari zat aktif saja. Saya yakin pengujian ini tidak banyak dilakukan di industri, saya cukup yakin karena ya paling banyak yang melakukan disolusi itu untuk QC gitu, pasti. Nah, yang berbeda dari semua jenis uji disolusi adalah surface area. Jadi, dalam uji disolusi intrinsik kita mengunci atau menetapkan atau menjaga luas area permukaannya itu konstan. Sehingga faktor-faktor lain selain sifat intrinsik dari bahan tersebut akan tidak berarti, hampir tidak berarti. Gitu. Jadi betul-betul melihat sifat intrinsik atau karakteristik dari zat aktif yang kita punya gitu. Nah, berbeda dengan disolusi lain pada umumnya, IDR bukan Indonesian rupiah ya, tapi intrinsic dissolution rate itu dinyatakan dalam bobot per sentimeter persegi per menit atau per detik. Umumnya menit lebih digunakan gitu. Jadi biasanya miligram atau mikrogram per sentimeter per menit gitu misalnya. Ya. Nah, masalah yang dihadapi di industri farmasi di Indonesia banyak yang melibatkan bahan baku. Ya. Ganti source. Uji BE mungkin, uji stabilitas gitu. Ini bisa nggak sih saya ganti source begitu saja? Kebanyakan kan tidak bisa ya. Dalam kasus dan kebanyakan terjadi masalah gitu. Kok sebelumnya BE, kok sebelumnya F2-nya bagus, kok jadi tidak? Itu salah satu masalah yang sering saya dengar di industri. Ya. Nah, ini yang mungkin akan saya sedikit berikan atau share. Mungkin kita bisa gunakan uji ini untuk melakukan troubleshooting dalam hal itu gitu. Nah, ini ya. Jadi raw material yang ada di secara komersial ya di pasaran itu biasanya memiliki kalau kita bicara supplier yang berbeda, kemungkinan memiliki jalur sintesis atau kristalisasi yang berbeda. Kalau orang-orang yang sering nyari source tuh pasti tahu, baca dokumennya, kemudian lihat pelarutnya beda nih di sini. Jalur sintesisnya beda nih gitu. Nah, kemudian particle size-nya juga bisa berbeda. Kemudian ini ya, kalau kita punya suatu bahan aktif yang hidrat, misalnya bla 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 monohidrat, mungkin derajat hidrasinya berbeda. Dan habit atau bentuknya seringkali juga berbeda ya. Tampilannya, morfologinya juga beda gitu. Itu masalah yang sering ditemukan ketika mencari supplier lain. Dan jangankan supplier lain, batch yang berbeda di supplier yang sama pun bisa memiliki sifat-sifat ini yang bervariasi gitu. Itu kan ya masalahnya gitu. Nah, ini yang nanti akan kita lihat bagaimana uji dissolusi intriksi itu bisa melihat perbedaan itu gitu. Nah, ini adalah contoh hasil uji dissolusi yang pada umumnya dilakukan ya. Dissolusi dari tablet ini adalah contoh data dari tugas akhir mahasiswa saya. Dia melakukan uji dissolusi tablet di Kagleror misalnya. Ini tipikal data uji dissolusinya seperti ini ya. Nah, untuk data intrinsic dissolution itu datanya bentuknya seperti ini. Jadi datanya adalah data yang linear. Jadi seperti garis regresi linear ya. Berbeda kalau uji dissolusi kan umumnya ada sisi apa istilahnya? Seperti logaritmik ya. Nah, tapi kalau IDR itu datanya linear seperti itu. Kemudian yang jelas juga adalah lihat waktunya. IDR itu cukup dilakukan dalam hitungan mungkin 10 menit selesai eksperimennya. Itu adalah contoh bagaimana simetidin bisa dilihat laju dissolusi intrinsiknya pada pH yang berbeda gitu. Jadi kelihatan dia paling bagus di pH 1 misalnya karena dia pK-nya 6,8 ya otomatis lebih larut di pH yang lebih asam dan paling rendah di pH yang mendekati pKa-nya. 6,8 jelas ya, berarti 50% yang terionisasi kelarutannya rendah gitu. Ini contoh dari data uji dissolusi intrinsik. Dan ini salah satu paper yang dirilis oleh Lawrence Yu ya kalau tidak salah. Ini juga saintis yang kemudian ke FDA nantinya. Sekarang udah sepertinya masih di FDA gitu. Nah, ini adalah salah satu penelitian kami di mana uji dissolusi intrinsik ini bisa membedakan kelarutan dari berbagai zat aktif yang sama yang dimodifikasi melalui pendekatan yang berbeda gitu. Jadi di sini ada polimorf dan juga ada kokristal ya atau garam lah bisa dibilang. Jadi kita melakukan perubahan dari sisi bahan baku, kemudian kita lihat laju dissolusi intrinsiknya. Nah, pertanyaannya kenapa tidak melakukan uji kelarutan saja? Kenapa kita melakukan uji dissolusi gitu? Ada kan ya? Mungkin yang pernah praktikum gitu ya di kelas atau di ketika kuliah pasti mungkin pernah melakukan uji kelarutan gitu. Dimasukkan ke dalam Erlenmeyer, kemudian dimasukkan ke dalam water bath yang diaduk atau digoyangkan selama 24 jam biasanya. Nah, ini perbandingannya. Jadi misalnya uji dissolusi intrinsik itu lebih dekat korelasinya dengan in vivo karena berhubungan dengan kinetik bukan kondisi termodinamik. Kalau uji kelarutan kan kondisi jenuh ya yang jarang tercapai gitu dalam kondisi nyatanya. Sementara kalau uji dissolusi intrinsik ya seperti uji dissolusi dalam kondisi yang sing yang mirip dengan kondisi in vivo. Kan ketika in vivo itu ketika yang sudah larut langsung diabsorpsi ya, diserap gitu sehingga dalam kondisi di situnya itu tidak dalam kondisi jenuh gitu. Kemudian durasinya tadi sudah saya sampaikan hanya 10 sampai 30 menit, cepat. Kemudian sampelnya sedikit. Jadi kalau uji dissolusi powder perlu mungkin di atas 100 mg, kalau uji dissolusi intrinsik bisa di bawah 50 mg. Dan terakhir, karena uji solubility atau kelarutan itu lama, maka ada perubahan transformasi kristal ya, itu bisa terjadi selama proses eksperimen, tetapi kalau uji dissolusi intrinsik tidak, karena waktunya sangat singkat gitu. Jadi secara umum, intrinsic dissolution itu adalah seperti pengganti uji kelarutan yang bisa dilakukan dalam waktu yang singkat. Prinsip dasarnya seperti itu. Nah, bagaimana cara melakukannya? Ada 2 jenis alat uji dissolusi yang ada di USP. Rasanya di Pharmacope belum ada chapter khusus terkait intrinsic dissolution, seingat saya. Jadi ada 2, ada rotating disk atau istilahnya wood apparatus dan ada stationary disk. Wood apparatus bentuknya seperti ini dan ini adalah yang lebih umum digunakan. Nanti akan saya tunjukkan lebih detail. Dan yang kedua adalah stationary disk. Jadi bagaimana disknya itu apakah diam atau berputar. Disknya itu adalah disk yang akan berisi sampel gitu. Yang stationary kan disimpan di bawah seperti ini, jadi dia diam. Menggunakan alat USB tipe 2 untuk pedal. Tetapi kalau rotating disk kita menggunakan spindle atau shaft yang berbeda gitu. Istilahnya spindle tapi ya. Nah, ini adalah contoh alatnya ya seperti ini. Jadi ini adalah attachment yang bisa di-connect dengan alat dissolusi apapun. Jadi kalau sudah punya alat uji disolusi tipe 1 atau 2, pasti bisa membeli ini. Tinggal bilang saja gitu ya ke suppliernya, mau beli alat uji disolusi intrinsik misalnya ya. Saya tidak mau minta beli, tapi itu bisa gitu, sesuatu yang bisa dilakukan. Atau bisa membuat sendiri kalau memang mau gitu ya. Jadi secara eksperimen kita hanya cukup memasukkan serbuk, biasanya berupa bahan aktif, ke dalam dice-nya, kemudian dice-nya di punch atasnya disimpan ke dalam dice dan dilakukan kompresi dengan pompa hidrolik manual seperti ini juga cukup. Karena tekanan yang diberikan cukup sekitar 1 ton lah, sekitar. Itu jauh lebih kecil dibandingkan tekanan pengempaan tablet ya, biasanya di sekitar 5 ke atas biasanya pengempaan tablet. Ini hanya 1 ton, kemudian kita akan mendapatkan seperti ini. Jadi seperti tablet, tapi kita tidak menyebut ini tablet, sebutnya kompleks. Yang kemudian akan disimpan di shaft dan disimpan di alat uji disolusi seperti ini. Jadi prinsipnya seperti uji disolusi yang basket ya, berputar sampelnya, tetapi dibentuk dalam luas permukaan yang konstan. Jadi itu luas permukaannya 8 mm dikali itu kan diameter ya, berarti pakai rumus itu ya, 2 pi apa tuh? PR kuadrat lah. Luas permukaannya segitu. Sekitar 60 sekian milimeter persegi kalau tidak salah. Itu konstan selama pengujian. Nah, ini adalah parameter yang umum digunakan. Rotation speed-nya ini jauh lebih tinggi dibandingkan uji disolusi pada umumnya ya. Biasanya kan uji disolusi berapa? 50, 75, 100 ya, itu paling populer. Tapi kalau uji disolusi intrinsik itu sekitar 300-an. Bisa sampai 500 RPM, jadi cepat sekali. Volumenya bisa kita turunkan tergantung kelarutan dan minimum 5 sampling point ya. Gitu kan mungkin untuk membentuk profil yang konstan, yang bagus gitu ya profilnya, kelihatan. Nah, ini adalah contoh case tadinya. Jadi tadi sudah eksperimentalnya ya. Saya akan menunjukkan terus gunanya apa gitu kan ya. Yang penting kan itu dari semua ini. Terus apa yang bisa kita gunakan gitu atau yang manfaatnya apa untuk kita di industri misalnya ya. Yang pertama, ini contoh penelitian kami juga. Kami melakukan modifikasi dan uji disolusi intrinsik ini bisa membedakan antara garam free base dan garam yang bentuknya hidrat. Padahal ukuran partikelnya yang misalnya contoh ya, ini kan yang tertinggi dari garam hidrat, kemudian garam anhidrat, kemudian garam hidrat dan yang terakhir free base. Kalau kita lihat dari sisi ukuran partikel, yang tertinggi malah garam hidrat. Free base paling kecil kan ya? Tapi dia paling rendah disolusi intrinsiknya gitu. Jadi uji disolusi intrinsik itu akan menggambarkan kelarutan dari obat secara kinetik dengan menghilangkan faktor partikulatnya. Gitu. Jadi ukuran partikel itu akan diminimalisasi dari sisi pengaruhnya terhadap kecepatan pelarutan dari suatu bahan gitu. Nah, itu ya. Jadi itu yang bisa dilakukan. Nah, terus ini mungkin yang lebih relate setelah ini. Next. Gak bisa. Nah, ini ya. Jadi ada bahan aktif lain misalnya diklofenak. Ini hanya perbedaan antara hidrat dan anhidrat ya. Jadi sama-sama garam, tetapi disolusinya bisa terlihat berbeda sekali dengan menggunakan uji disolusi intrinsik. Contohnya di pH 4,5 gitu. Kelihatan. Kalau misalnya kita tidak sadar bahwa kita biasanya menggunakan yang anhidrat misalnya yang komersial, tapi ternyata suppliernya bentuk hidrat misalnya ya, yang terjadi ya masalah yang sering dihadapi di industri. Kok disolusinya tiba-tiba tidak masuk ya? Kok tiba-tiba tidak bioekivalen lagi ya? Nah, kayak gitu yang bisa terjadi gitu. Jadi ini adalah salah satu tools yang bisa digunakan untuk mencegah itu terjadi. Ya kalau kita punya bahan baku dari supplier yang berbeda, kita tes dulu. Misalnya, contohnya. Nah, selanjutnya yang lebih relate adalah yang ini. Jadi ini ada simvastatin BCS kelas 2 ya, yang masalahnya disolusi juga. Jadi researcher ini, saya tidak tahu ini iseng atau gimana ya, tapi intinya memodifikasi bahan tersebut dengan merekristalisasi bahan baku yang dia punya di pelarut yang hidrofilik dan hidrofobik gitu. Jadi bahan bakunya sama awalnya, antritid ini ya. Kemudian dia kristalisasi ulang di pelarut yang hidrofilik dan di pelarut yang hidrofobik. Jadi bahan bakunya betul-betul sama nih, simvastatin dari source yang sama. Nah, ternyata Mereka coba dengan kontak angle ya, jadi melihat sudut kontaknya yang lebih rendah, yang lebih hidrofilik ya. Lebih tinggi itu lebih hidrofobik. Dan ukuran partikelnya ya tidak terlalu jauh lah. Antara yang hidrofilik dan hidrofobik ya. Tidak terlalu jauh. 19 dan 21 itu tidak berbeda lah bisa dibilang. Dari segi XRD, kalau mungkin ada yang familiar dengan ini, lihat polymorphnya dengan XRD, ya sama aja kayak gini. Nggak akan terdeteksi dengan menggunakan XRD. Identik karena bentuk kristal atau polymorphnya sama. Hanya berbeda dari sisi modifikasi partikelnya saja. Tapi ternyata dengan uji disolusi intrinsik, terlihat perbedaannya gitu. Jadi sesuatu yang terlihat seperti sama, tidak berbeda, mungkin bisa terlihat berbeda dengan uji disolusi intrinsik seperti ini gitu. Ini. Nah jadi ini yang tadi saya bilang, ya kalau kita ganti raw material, misalnya ada di X perusahaan A, X perusahaan B, kadang kan ragu ya, atau perlu justifikasi khusus kan ya, kalau misalnya mau registrasi variasi atau apa gitu di Badan POM, ya ini bisa menjadi salah satu data yang bisa digunakan. Oh, di uji disolusi intrinsik sama kok, tidak berbeda signifikan. Itu misalnya ya, sehingga bisa meyakinkan misalnya orang formulasi atau orang produksi bahwa ini adalah bahan baku yang memiliki karakteristik yang sama. Nah satu lagi, terakhir ini, raw material QC ya. Jadi ada orang yang menggunakan 7 batch raw material yang berasal dari 3 different supplier ya. 3 supplier bahan baku yang berbeda. Terus ada 7 batch dari Evavirens BCS kelas 2 ya, sengaja dipilih BCS kelas 2 karena yang paling relevan dengan disolusi. Dan ternyata dengan mereka mendesain suatu uji disolusi intrinsik yang bisa membedakan ke-7 batch tersebut. Jadi mereka mendevelop sendiri dan ketemu bahwa dengan menggunakan SLS 1% di mediumnya, mereka bisa melihat perbedaan yang paling signifikan. Jadi contohnya yang batch A 71, yang batch B itu 59 dari segi mikrogram per menit per sentimeter segi ya. Jadi 71 dan 59 dengan standar deviasi yang kecil seperti itu kan berbeda signifikan ya. Jadi mereka bisa mendesain suatu uji untuk membedakan karakteristik antar batch. Terus implikasinya apa ya? Yang menarik adalah batch A itu adalah bioekivalen. Mereka menguji batch A dan batch B dari 2 sumber bahan baku yang berbeda. Yang satu bioekivalen, yang satu tidak. Dan itu terdeteksi dengan mendesain uji disolusi intrinsik. Dari sini mereka bisa menentukan kalau nanti suatu saat saya akan membeli dari source lain, maka cut off uji disolusi intrinsiknya adalah 39 atau 35 lah mungkin. Jadi kalau nanti ada supplier lagi masuk, tes uji disolusi intrinsik di atas 35 masuk, di bawah 35 ada resiko tidak bioekivalen. Ini salah satu yang mungkin bisa diaplikasikan. Jadi kan banyak ya tools yang bisa membantu kita. Ada XRD misalnya, uji disolusi serbuk. Nah, tapi nggak ketemu nih bedanya apa. Nah, mungkin ini bisa menjadi salah satu metode yang bisa diimplementasikan untuk menyelesaikan masalah itu. Nah ini ya, jadi kalau ada satu pesan yang diingat dan dibawa pulang gitu ya, cukup satu ini saja. Jadi uji disolusi intrinsik atau ya intrinsic dissolution rate itu adalah suatu tools yang bisa digunakan sebagai pada saat preventif dan juga troubleshooting untuk bisa mengakses ya, bisa menguji dan juga mengakses. Kita bisa mengetahui perbedaan karakteristik dari bahan baku gitu. Itu ya, jadi kalau selama ini misalnya pendekatannya adalah ya udah dibikin dulu baru diuji disolusi terbanding dan nggak ketemu kenapa bisa hasilnya tidak BE, mungkin kita harus kembali ke hulu. Lihat bahan bakunya lagi. Gitu. Karena ya walau bagaimanapun formulasi itu bukan satu-satunya faktor yang akan mempengaruhi performan dari produk kita gitu. Bahan bakunya terutama API dan juga proses ya, ada lagi prosesnya nanti. Tapi ya ketiga itu mungkin harus diakses dan salah satunya untuk mengakses raw material kita bisa menggunakan uji disolusi intrinsik. Itu saja dari saya. Thank you very much for your attention. Good afternoon everyone.
Okay, thank you, Dr. Yuda, for your presentations. And now we are move on to our next sessions. We like to invite Mr. Suhasyo Wale as Associate Director of Techno Commercial Sotex India who will be presenting on method development and release applications using the USB Type 4 apparatus. Please welcome Mr. Suhasyiwale. Here, Mr. Suhasyiwale, as a seasoned analyst, R&D professional with an Magister Science in Analytical Chemistry from Mumbai University and over 30 years of experience in senior management roles at leading generic pharmaceutical companies such as Sandoz, Glenmark, Famicare, and Par Formulations. Currently, Mr. Suhasyiwale is serving as Associate Director Techno Commercial at Sotex, India, and overseeing the Dissolution Application Lab at Bombay College of Pharmacy.
Hello. Yeah. So, so good afternoon, everyone. Yeah, all are waking up. Good. After having a lunch, it is difficult for me to keep you awake, but I will try my best to make you interesting. So I'm very thankful for the School of Pharmacy, ITB, for giving me this opportunity to present dissolution by USB Type 4 and its applications. Next slide. Can you slide changer? So in this presentation, I will tell you about the introduction, method development, release application of USB Type 4 apparatus for dissolution testing. Next slide. What we are going to see first, what is dissolution? What is the dosage form challenges that we face when we do the dissolution method development? Or the third is the comparison of USB Type 1 and Type 2. Most of you are used Type 1 and Type 2 many times, but Type 4, what it is and how it is comparable with Type 1 and Type 2, that I will explain you in brief. Then what is the open loop and closed loop system? Here we have kept the apparatus for demonstration. After we finish this, we can explain you what is type open loop and what is closed loop. Then smart development, right from API to finished dosage form. We can use very effectively this apparatus for your API dissolution and the finished dosage form. You name the dosage form, you can use this USB Type 4 for your dissolution testing. Okay. Then what are the analytical configuration that Sotex is offering for USB Type 4 apparatus and its applications? I will go through some applications for this USB Type 4. Next slide. So basically there are two types of dissolution. When we do in a dissolution apparatus, we call in vitro dissolution. And when a dissolution happening in your body, it is called in vivo dissolution. In a simple word, dissolution is nothing but kinetics of the API released from the dosage form. Okay, as simple as that. It is the kinetics of the API that is getting released during dissolution testing. And when we do in vitro dissolution, like apparatus 4 or apparatus 2, apparatus 1, we get a cumulative curve when it is a closed system. In a fixed volume of the dissolution medium, we get a cumulative curve. But when we do a in vivo dissolution, we get a differential curve at absorption and elimination phase that you can see from the picture here. It reaches to a maximum, and after that excretion of the drug starts, which in a closed system will not be able to see because you get a cumulative curve. Next slide. So what are the challenges that we face when we are doing dissolution testing or dissolution method development using type 1 and type 2 apparatus? Okay. So here in this slide, you can see these are the different dosage forms and what kind of challenges that we will face during dissolution method development or dissolution testing. So coming to the first dosage form, that is modified release or extended release tablets, the challenges that we face is the pH change. Many times for extended release or delayed release tablet, we need to change the dissolution medium. First in the acidic condition, followed by a phosphate buffer pH 6.8. So if you are doing that apparatus 1 and 2, that time you will have to stop the dissolution after you finish your 0.1 normal HCl dissolution, and then you have to remove that medium by decanting it, and again you have to introduce another medium in which you want to study. Okay, so that is interruption of the dissolution is happening. But in open loop system, you can do without interrupting, without stopping the dissolution. That is the advantage. Then poorly soluble. If your drug is poorly soluble, like BCS class 4, to achieve the sink condition, there is a limitation with apparatus 1 and 2. If you want to use more than 1 liter, or there are apparatus which are having 2-liter vessels also, but still your drug is BCS class 4, it is insoluble in aqueous medium, that time you will need more volume of the dissolution medium to get the sink condition. So that time we can use open loop system, and infinite volume of the dissolution medium can be passed through that dosage form so that you can get the sink condition. So this is one thing, these challenges that we can overcome by using type 4. It is not that only type 4, with type 1, type 2 are also we can overcome that by adding some surfactant. But if you want to truly see the aqueous medium in the body, how it functions, that we can use type 4. Then coming to the second thing, that is API or the powder or the granules. The challenges that we face is the low dose and hydrodynamic control. Because of that API powder, it gets floated if you use apparatus 1 and 2 as it is. Just previous presentation, Dr. Yuda has explained you intrinsic dissolution. You can use intrinsic dissolution also. But in apparatus 4, there is a separate cell that is called powder cell. We can use that powder cell and you can do that dissolution without making a compaction or without using that another assembly for your apparatus 2. Then coming to the third dosage form, that is capsule and the pellets, where you many times face a floating problem and the capsule interference. Here in apparatus 4, we can overcome by using apparatus 4 with the specific cells, and this floating can be, these interferences due to the capsule cell can be avoided. Then coming to the medical devices like drug eluting bed, stents, or the implants, or these coated ocular implants, again, there is a challenge of low dose. Most of the time, these medical devices are having a drug which is in microgram level. The another challenge that we face when we come with apparatus 1 and 2 with 900 ml of the dissolution medium is the analytical sensitivity of the dissolution sample that we are testing in the HPLC. So with apparatus 4, we can reduce the volume to even 25 ml. So you can do that type of testing using type 4. Then coming to the suppositories and soft gelatin capsule, the interference due to the lipid interference. That is the problem that in apparatus 4, we have a twin chamber cell where there are two separate chambers. You have to keep that suppository in one chamber. The dissolution medium comes in contact with that, and the lipidic residue that comes out will get settled on the top of the cell, and you will get a clear liquid which is coming out from the middle of the cell. So that interference can be avoided. Last but not the least is that parenterals, emulsions, suspensions, microspheres, and the semi-solids. The challenges, a lot of challenges are there. First thing is the low dose. Second, small particles. And many times dialysis is often required. USP type 4 is the compendial apparatus which can be used for all these dosage forms. If you want to do dialysis, apparatus 1 and 2, you cannot you can do some experiment, but to have a compendial apparatus, then you'll have to use a dialysis membrane. There is a special adapter. We call it as this type 4 A4D adapter, which is used for the dialysis membrane. And there are some ready-made uh, dialysis tubes are available that is called floatalizers. Different molecular weight cutoff. These floatalizers are available. You can put your drug product into that tube and you can perform the dialysis using apparatus 4. Next slide. Now we'll see the comparison. Next slide, please. Yeah. We can see the comparison of apparatus 1 and 2 with apparatus 4. There are 4 different columns I've made. The first column, where on the basis of these various parameters, we can compare apparatus 1, 2 with open loop and the closed loop system. Coming to the first point, that is dissolution volume. In apparatus 1 and 2, it is constant. Even in closed loop system of type 4, it is constant, say 900 ml or 1 liter or 2 liter. But in case of open loop system, in finite volume, it is continuously flowing, that fresh dissolution medium flowing through the dissolution cell or your dosage form, and it is a dynamic, continuously fresh medium is going. Whereas in type 1, type 2, the same medium is getting circulated in closed system, and 1 and 2, the constant volume. Coming to the second point, dissolution concentration. Or you can see the last row where we can say dissolution profile. In that case, closed system and the type 1, type 2, since it is a constant volume, we get a cumulative curve. When we do a dissolution profiling, then we get a cumulative curve. But in case of open loop system, we get a differential curve like in vivo, where the drug reaches to a maximum and the excretion starts. Similarly, you get a differential curve because in open loop system, fresh medium is going inside the cell and it is coming out and it is not going back into the cell again. The excess amount goes to the waste and we collect a fraction of it for testing the drug dissolve in that particular volume in that particular time. That can be studied. Then agitation. For any dissolution test, agitation is very important to get a drug release from that dosage form. So in case of type 1, type 2 apparatus, the agitation is because of the speed of the paddle or the basket. We fix that particular speed, 100 RPM or 50 RPM, and we get agitation and the drug gets released from the dosage form. In case of USP type 4, the agitation is because of the flow rate. By using this apparatus, we can have a variety of or a maximum option of agitation by putting 1.5 ml, the minimum flow rate of 1.5 ml to the maximum of 35 ml. That much options are available to get the agitation in the dissolution testing using USP type 4 apparatus. And the another phenomenon is evaporation. You must have heard this evaporation in apparatus 1 and 2. Suppose your extended release tablet or sustained release tablet and your dissolution is of say 48 hours. That time you started with your dissolution test with 900 ml, and over a period at 37 degree, at the end of 48 hours, if the lid of the vessel is not properly closed, or there is a chances because of the evaporation of this medium for such a long time, you will find the volume of the dissolution medium is going down. From 900 ml, it may go to 850 or 875 ml. So it is changing the volume. But in case of open loop system or a closed loop system, there is no surface, media surface get open access. So there is this evaporation phenomenon is not observed in case of type 4 apparatus. And configuration, as you can see, this apparatus 1 and 2 and the closed loop system, they are in the constant volume, they are more or less same. But in open loop system, a continuous medium is passing through your dosage form and it doesn't come back, it goes to out of the cell. So next slide, please. So we'll understand the mechanism of open loop system. So here, a schematic diagram to understand this, what is meant by open loop system, how it works, how it functions. Okay, you can see here, can you see this side? The first is the 3 media. So using apparatus 4, you can do a multimedia dissolution without stopping the dissolution. At any given point of time, you need not to stop the dissolution till you complete your 4 different media. And after this presentation, I will show you how it works on the apparatus also. What is this? Loose connection? Can you go back? That LED. Okay. Then after that, 4 reservoir, you can see here also, we have kept 4 reservoir for the dissolution medium. And after that, there is a media selector. You'll have to program your dissolution method in such a way that 4 different media. Suppose you want to start with 0.1 norm HCl, followed by pH 4.5 acetate buffer, followed by phosphate buffer pH 6.8, and followed by pH 7.4. So you will program, suppose you want 0.1 norm HCl for 2 hours, you can program it accordingly in the method that you want 0.1 norm HCl 2 hour, pH 4.5 2 hour, pH 6.8 for 4 hour, and pH 7.4 for 8 hour. Accordingly, you can plan that and it will run continuously without stopping at any given point of time, and you will get the complete profile, entire GI tract, how your drug is getting dissolved. You will get the complete picture of that. Then comes the pump, very precise, accurate, 1.5 ml to 35 ml. It will work, and there are 7 individual pumps for the 7 cells which is kept in the dissolution apparatus. From that pump, the dissolution medium passes to the main cell which is fitted in the dissolution apparatus. Like apparatus 1 and 2, there are 7 cells, 7 vessels. The 6 vessels contain your dissolution medium and the drug product, and the 7th cell is for the replenishment, or you can put the placebo. Similarly, here also there are 7 cells, 6 cells, you put your tablet or the capsule or the any dosage form that you want to test, and the 7th cell you can put either placebo or the plain API. If you want to see how my plain API behaves in as it is without the formulation, that also you can see. Then comes the sampler, or we can say the splitter. When the dissolution passes here in open loop, as I told you, the dissolution medium will pass over the dosage form and it will not come back again to the cell. So how I will test that which is getting out of the system? So the function of the splitter is that it will split that output of the dissolution medium which is passing through the dissolution after testing, after dissolution, it will get split it into two parts. One part will go to the waste and the another part will go to the fraction collector test tube, which is a flat bottom 65 ml capacity flat test tube. Now I will explain with a simple example how it works. Hope you understood the mechanism how it works, but I will explain you how that splitter will function. Suppose your flow rate is 10 ml per minute and your first sample is coming at 60 minutes. You want to test that at the end of 60 minutes. So flow rate 10 ml multiplied by time point, that is 60 minutes. So output of the dissolution medium will be 600 ml, 10 multiplied by 60. But the capacity of the fraction collector test tube is only 65 ml. You cannot collect 600 ml. So I will program the method in such a way. For analytical testing, I will need hardly 20 ml or 30 ml or 10 ml, whatever ml. Suppose I collected 20 ml. So 20 ml will get collected into the fraction collection test tube and remaining 580 ml will go to the waste. And at the end of 10 minutes, 60 minutes, this dissolution collection, output collection will start from the minute 1 till the 60 minutes. So you will get a representative sample for that 60 minute, entire 60 minutes, starting from 1 minute till 60 minutes. Similarly, you can program like after 1 hour, after 2 hour, 4 hour, automatically it will get collected and the excess volume, suppose the entire 8 hour, you are passing say 50 liters of the dissolution medium. And every time point you want to 20 ml, you can program in this way, 20 ml will get collected into that particular time point and the excess medium will go to the waste. I will show you that in the dissolution apparatus also. So next slide, madam. So what is the advantage of this open loop system is that it maintains the same condition for poorly soluble compound like this is class 4, where you get a low solubility. Then it is easy to operate to change over the pH dissolution medium, different dissolution pH medium. You can, without stopping that, you can use it. Next slide, please, madam. Now, I will tell you about the configuration of the closed loop system. Hope you have understood the open loop system, correct? Okay. Still, there is time who has not understood. I will show you on the apparatus directly, okay? Here, in this closed loop system, as from this picture also, you can see it similar like apparatus 1 and 2. But it has more advantages compared to type 1 and type 2. So that it is flexible with small and the large volume. Okay. Then both the small and the large volumes of the dissolution media can be used without changing the hydrodynamic conditions experienced by the dosage form. Here, as less as 15 ml I can use, and as high as 400, 4000 ml, that is 4 liter of the dissolution medium can be used in the closed loop system, which is not possible with apparatus 1 and 2, where the vessel minimum volume you will have to use 500 ml. But here I can go up to 15 ml also. Okay. For dosage form like ophthalmics, how much? 100 ml. Even a person cries for 8 hours, he cannot collect more than 100 ml. Correct, sir? So this is ophthalmics. Generally, we do less volume of the dissolution medium, artificial tear fluid. Okay. Then it is ideal particularly for the low dosage form. Suppose you are having a potent drug for a contraceptive hormone like where the dosage form has 0.1 milligram. If you are doing 0.1 milligram in 900 ml, very sensitive, your drug dissolution, very difficult to test by using even HPLC, then you will have to use special detector like fluorescence detector and all that. But with the small dosage form, you can use a cell which is having a 22 mm cell where 14 ml will be exposed, and the reservoir can be of 100 ml or 500 ml. Okay. So this is the closed loop. Next slide. Now I will tell you the more advantages of closed loop system over type 1 and type 2 apparatus. So in pharma industry, all of you are using type 1, type 2 apparatus for immediate release dosage form. But type 4 has more advantages. Which are those advantages? First thing is reproducible position. You can see that cell where the tablet is placed on the hanger, tablet hanger, where the flow through cell allow for the reproducible positioning of the dosage form and eliminates many problems like sticking or the floating of the sample problems experienced during apparatus 1 and 2. The second advantage is flow rate versus the stirring speed. Where you, in type 4 apparatus, there are a lot of like 2 ml, 4 ml, 8 ml. As per USP General Chapter 711, they have given 3 standard flow rate, 4 ml, 8 ml, and 16 ml. But here, as per your requirement, as per your expected dissolution profile, you can change from 2 ml, 3 ml, 4 ml, 8 ml. Lot of variety options are there. Then expanded operating volume. Apparatus 4 can be run with dissolution volume below, at, and above the capabilities of the apparatus 1 and 2. Below means less than 500 ml. Apparatus 1 and 2, you cannot run with less than 500 ml. But here I can run below 500 ml, 100 ml also I can run. Then add means 900 ml or maximum 2 liter. I can run closed loop system with 2 liter also, even 4 liter also. And above means above 2 liter. So this is the advantage that we get. Then different cell types and with the glass bead or without the glass bead. You can see in that cell, the small glass beads are kept, 1 glass beads. You can run with glass bead and without glass beads. So in a nutshell, I will tell that apparatus 4 has more discriminating options compared to type 1 and type 2 apparatus. And it is very important that any dissolution method that you develop either for your development purpose, product development purpose, or it can be a release purpose in the quality control, it should be discriminating. And if your immediate release drug product is highly soluble, highly permeable, and it is getting in all the medium very fast without showing any discrimination, you can use apparatus 4 with the closed loop system to show the discrimination. Okay? Many times FDA is asking for the discriminating dissolution nature. That time, apparatus 4 will be very useful for you. Okay. Next slide. So this is the picture of open loop system where the basic dissolution unit and the pump remains the same. This is the media selector which has 4 channels to run 4 different media. Then that is the fraction collector with the splitter to split the dissolution medium into 2 parts. One goes to the test tube, another goes to the waste. And another is the closed loop system where this dissolution unit and the pump remains the same. The fraction collector changes with the syringe module, where you can collect directly into HPLC vials the amount of that dissolution medium that you want to collect. If you are using UV spectrophotometer, you can collect 5 ml, and if you are using HPLC, you can collect 1 ml of the dissolution medium that is coming out of the cell. Okay, next slide. Smart development. For smart development, here apparatus 4 provides different cell types and different adapters for your different dosage forms. Generally, with apparatus type 4, Sotex is providing 2 types of cell. One is a small cell that is 12 in diameter, specifically for the low dose tablets, 5 milligram, 2 milligram tablets, and a large cell with a 22.6 diameter. Okay, so these 2 cells are generally provided, but this 22.6 cell is a multi-purpose use with different dosage form. Like if you want to do a semi-solid oil, ointment, cream, or gel, then we provide a semi-solid adapter, and you can use the same cell for your that type of products. Even for suspensions or emulsion, where you need a dialysis, you can use this dialysis adapter. Okay. Then for medical devices also, we have a 22.6 where we have a different adapter for the ocular lenses and all that. Coming to the fourth cell, that is a powder cell. In my previous presentation, Dr. Yoda has told you about the intrinsic dissolution. In my next slide, I will show you what is the difference between the intrinsic dissolution and the apparent dissolution. I will explain you in more elaborately. Then this is the suppository cell. It has two chambers. In one chamber, you can place your drug product, and another chamber where the clear liquid comes, and from the center of that, it goes out. Then there is a medical devices, three cells. Next slide. Yeah. So here we'll understand what is intrinsic dissolution and what is apparent dissolution. As Dr. Yoda has very well explained you, and the importance of intrinsic dissolution, I will tell you when you, most of you are studying your post graduation or the PhD, when you go to industry, if you do this biopharmaceutical characterization of your API before starting your drug product development, I will tell you it will reduce your most of the development time. Why? Because I will tell you like this. See, what is the difference between the intrinsic dissolution and apparent dissolution? As per USP General Chapter 1087 or the European General Chapter 2. EP General Chapter 2.9.29, intrinsic dissolution can be done by two apparatus, two methods, rotating disc or a stationary disc. You must have seen in the earlier presentation, where you have to compact your API at maybe 2 pound pressure for 2 minutes or 1.5 pound pressure for 2 minutes, and a fixed surface is exposed to the dissolution medium, and you get. But sometimes it has some disadvantages for the API which are fragile in nature. The crystal, as all of you know, that dissolution is directly proportional to the particle size and the morphology of the API. Suppose your API is having a needle shaped morphology and it is little bit fragile. That time what will happen when you compact it into like a KBR pellet, you compact it at 2 pound pressure, there is a chances of breaking of that needle. So it becomes just a solid state material. And when we do that dissolution, you find a difference in the dissolution profile, or different solvents or different hydrates or two different polymorphs. You can see in this the form 1 and form 2. When it is done by intrinsic dissolution, it is showing the same dissolution profile because it has broken into two parts like a solid particles only. But as per the EP General Chapter 2.9.43, apparatus 4, where you can use the powder cell to perform the dissolution of the API. Here, we can put that API without compacting into a disk. As it is loose powder, let it be any needle shape or the spherical shape or the plate shape API, you have to just put the powder as it is a weight quantity into the dispersion disk kept inside the powder cell. And the same form 1 and form 2, you can see the dissolution profile. They are showing two different dissolution profile. Why? Because during the compaction, the particle has broken down and it has become a small solid state particle, and dissolution is directly proportional to the surface area of the particle. Smaller the particle, faster will be the dissolution. So this is how it works. Next slide. Now I will explain you how it works. Here, this powder cell, madam, can you one more time? It is divided into three parts. The lower part, which is shown in the yellow color, has two fixed of 450 micron SS sieve or 450 micron. In between these two sieves, we have to fix one filter paper, maybe of 2.7 micron porosity, that is a glass fiber filter paper. The function of this paper is that whatever API we put into the middle portion, it should not get escape or go below down the tubing. Okay. Then, madam, can you press again? The middle portion, which is shown in the green color, which has a dispersion disk where you can put your API, weight quantity of the API, 50 milligram, 100 milligram, depending on the nature of your API, fluffiness or the crystalline nature or the amorphous nature, the quantity of the API, weight quantity of the API, 50 milligram, 100 milligram, you can put into that dispersion disk. And above that disk, there is a blocking ring. That blocking ring, function of that blocking ring is that when the dissolution medium passes from the bottom of the cell, because of the flow rate, the disk will get lifted up. But the blocking ring will not allow that dispersion disk to go up top. Inside API of the dispersion disk, the API inside will get explode into that cell, and the dissolution will happen in that cell. Okay. Then, madam, you can press it. Oh, okay, it is already there. That blue color, that is the upper, that is the third portion, which is showing again the fix of 450 micron, and above which we can fix two more filter paper, 2.7 and 0.7 micron, so that not a single particle unfiltered or undissolved will pass out of the cell. That is the function. Madam, can you put I will show a small video here in that where two API size particles, no, back here, or I can use that. I will try to start that video. Can you put that video? There is on that picture, there is an arrow. See if you can on that picture. Not possible? Okay, sorry. So in that two cells, we have taken same API. One is a API is a coarser size, maybe D90 particles. Cannot run. Okay. Okay, fine. So what happens? You run. Okay. Started. Thank you. See, the left side cell has coarser particle and the right side cell has a
Et l'autre, il est parce qu'elle est plus fine, la poudre. Et l'autre aussi, il est passé à travers, ça a bien marché, tu vois. Et l'autre, il a complètement démoté, et là, par cet amour, il se passe plus rien. On voit juste qu'il... C'est pas mal, hein? T'as vu,
Il se dissout bien.
Ouais, mais l'autre, je mets le doigt dedans, moi.
Il se fait bien dissoudre.
Peut-être que j'ai mis le doigt devant.
You can see the dissolution pattern or the dissolution pattern in the cell, which is not possible with the restricted dissolution apparatus. That is the biggest advantage that you will get, and you will get the actual, the morphology, what is the morphology, what is the effect of that morphology on the dissolution rate. That is why I insist, before starting your product development, you do the complete biopharmaceutical characterization with respect to particle size and the dissolution profile. These two things are very important. When we go into the industry, please do that. Nowadays also, till date, some of the formulation scientists are not following this technique, and they land up into redevelopment formulation. Okay. So to avoid that, you use this and you will get a benefit out of it. Next slide. This is the suppository cell. Here you can understand the mechanism. Madam, can you just enter it? The blue dots from that, put enter. Oh, here again it is not working. I don't know. So as I explained you, there are two chambers in that cell. Right side chamber, there is a suppository we have kept, and you can see a angular tubing from which the dissolution medium will enter into that chamber. It will come in contact with the suppository and the lipidic residue that will go and sit on the filter. There is a the triangle one. and a clear liquid will come from the second chamber. And if there are in suppository, many times we use oil in water type of emulsion. That time some globules will be there. The pin will break that it will puncture that globule, and suppose there are some gelatinous mass which is coming out, there is a mesh below that which will not allow that particles to come out of this cell from the middle of them. So uninterrupted means no interference due to the lipidic residue, you can get a very clear dissolution medium out of that. And I have seen some of the research papers where apparatus 1 basket has been used for the suppository, where they are getting at the end of 60 minutes drug release maybe 10% or 20%. But when they have used this, they got more than 45% or 50% drug release, and very, very good discrimination compared to type 1 apparatus and the type 4 apparatus in the type 4 apparatus. Next slide, madam. So it is started. Yeah, good. See that blue dots are coming through that, and from the middle portion, from that filter cap, it is coming out. So you can measure very clear liquid which is coming out of this cell. No interference due to the lipidic residue. Understood? Okay. Next slide. So here again, some medical devices. We can use this cell and you can run this uninterrupted 60 days a dissolution without stopping a minute. Such a nice pump, very efficient and very precise. Okay. Next slide. For the microspheres, as all of you know that the microspheres, sometimes it takes days or weeks or sometimes months to get drug release from these microspheres. But with USB type 4 apparatus, where at accelerated temperature 45 degree or 55 degree, you can perform this experiment and you can reduce this a month's time to a 1 week time. Okay. And provided you have to show that it is a discriminating. And in my application, I will show you one application where FDA has accepted that as a release testing in QC. In QC, I cannot wait for a 60 days to get drug release. My product will not go in the market for the next 2 months. So I can do the accelerated dissolution and I can reduce the time, but provided I have to give that discriminating nature at that temperature. Okay. So next slide. Here, as I explained you, you can use the dialysis for the emulsion like liposomes. This is the structure of liposome, not going into detail. This is the ready-made flotalizers of different molecular weight cutoff or Moko value that we get. And you can put your emulsion directly into that and run the dissolution using 22.6 mm cell with a separate cap, filter cap, where you can put that because of the length of that flotilizer is bigger. We cannot use regular cap. There is a special cap for this flotalizers. Or you can use this USB type 4 A4D adapter for dialysis. It is reusable. Sotax will provide you this adapter. You have to wrap the this membrane and you can use repeatedly. Okay. So it is cost saving and time saving. Next. Here, semi-solid. As per USP General Chapter 1724, there are 3 methods described for this semi-solid product. First method is using by France diffusion cell. Second is by using apparatus 2 using the enhancer cell. And third is USB type 4 apparatus using the semi-solid adapter. Here, advantage is that orientation of the membrane in 2 different direction is possible. The way it is kept, the membrane is sitting on the bottom of that adapter and it is facing directly the flow of the dissolution medium. So this is, if you keep it upward, then the dissolution membrane is positioned in indirect flow, that is upward. So depending on the nature of the drug release or discriminating nature, you can put it in this direction or in that direction. So that is the benefit of that. Okay. Next. And it is available in 3 different sizes. This is a smart development right from the API using the powder cell, or some of your finished product may be in the form of granules or powder for solution. You can perform the dissolution in this apparatus, powder cell. And finally, the dosage form, the tablet or the capsule, you can perform using the tablet cell. So as per the principle of quality by design, first you will have to understand what is the nature of my API, what is the dissolution profile of my API, and what is the intermediate stage that I get, whether it is to process it further, I will first do the intermediate stage dissolution, and then I will go for the further test. So based on quality by design, a smart development using apparatus 4, you can do, and you'll get many, many advantages compared to type 1 and type 2. I'm not saying that it is to be replaced by apparatus 4 to be replaced by apparatus 1 and 2, but it has many options, discriminating options to use. Okay. During your development, I will say each and every company should have this for their development, which will reduce their development time, and they will understand the dissolution profile, which is very, very important performance test for your dosage form. If you study this API and finished formulation, which has more discriminating option in apparatus 4, and it is very easy to correlate your in vivo and in vitro dissolution by open loop system. Okay. Next slide. So analytical configuration, Sotex is providing this. Madam, can you put it again? Yeah, it is available with offline and the online. With the offline, that means you will collect the sample in the autosampler, or you can replace that autosampler or the fraction collector with the UV spectrophotometer where direct reading can be taken. Okay, that is the automation. And or you can have either both. Next slide, please, madam. Similarly, for this closed loop system also, there is, you can use with the fraction collector or with the UV spectrophotometer online. Next, madam. Okay, now I have completed my USB Type 4 instrumentation part. Now let us see some interesting applications. Okay, next slide. This is a example from US FDA OGD, Office of Generic Drugs, where you will get lot of dissolution examples using USB Type 4. Okay, this is a medroxyprogesterone acetate injection suspension, 150 milligram per ml, where this suspension initially OGD has suggested type 2 apparatus with 0.35% SLS in 900 ml and using 50 RPM. And the dissolution time was 2880 minutes, that is 48 hours. I don't know in 48 hours why they were doing, but actually when it was done, it was showing in 1 hour entire dissolution. But still it was. But then OGD realized that, and in the year 2014, they came up with the type 4 apparatus dissolution method, which is mentioned here. Type 4 flow through cell using 22.6 mm, 13 gram of 1 mm glass bead, 17, see the flow rate, 17 ml per minute. Very odd figure, correct? Standard flow rate is 16 ml, 4 ml, 8 ml, but they have 17 ml to get a discriminating dissolution profile. Okay, and 0.5% SLS, and you can see it is just 90 minutes, 90 days depot injection, and for QC release purpose, it is completing in 90 days, 90 minutes. See the difference. You need not to have your product hold up for 90 days in your quality control. You can release it in 90 minutes. Next slide, please, madam. You can see here the cell design, where the white color powder in that cell is the actual drug, 150 milligram of the medroxyprogesterone acetate, which is a contraceptive injection, which is placed sandwich method. They have mentioned use 13 gram. They are used 13 gram. They have mentioned, see, glass bead 13 gram, 1 mm glass bead, And they have mentioned all these conditions. OGD method gives maximum number of dissolution condition, but they don't give completely. That you will have to, for type 4 apparatus, you will have to optimize the condition to get a reproducible dissolution. So here we have used the filter, you can see GFD and the glass wool. What is GFD? There are glass microfiber filter paper. GFD, the D denomination has 2.7 micron. And there is another GFF, which is having 0.7 micron. Okay. So here we have used glass wool. In addition to the glass fiber filter paper, we added glass wool to prevent blocking as well as to get the reproducible dissolution. That was optimized in our lab. And you can see 2 runs of 6 units each, we can get RSD less than 2%. But when we started the dissolution, initially we were getting variation in the release profile. 1 or 2 cell was showing 75% release, some 2 cells shows 85% release, and another 2 cell shows 94% release or 79% release. So different dissolution pattern. But when it was optimized, see, OGD will not say what type of method or optimization of your cell preparation you'll have to do. That is why I say apparatus for dissolution is an art and the skill. Cell preparation is very important, very critical job in apparatus 4, where you need a proper planning, a designing of your cell in such a way that you get a reproducible dissolution. Okay. So that is the thing. Next slide. Then this is an ophthalmic emulsion where this floatalizer, that is the floatalizer was used using this closed loop condition where it is a 20-hour dissolution where the test and the RLD was performed and it has shown closely similar drug release, and the F2 value, the similarity factor was more than 70. So they got a confidence that it will pass in vivo also. Okay. Next slide, please. Here, the optimization has to be done. Again, I'm telling you, type 4 is not type 1, type 2 like simple dissolution apparatus. It needs skills and the art, both. You'll have to optimize your method. Whereas in apparatus 1 and 2, only volume and the RPM. You have to just drop your product in the vessel and forget it or observe the thing. But here, you have to work out. Okay. Here, first, this emulsion was directly put onto the glass bead inside the cell. You can see the orange color line. Within 5 minutes, more than 95% drug got released. But when we use the floatalizer, because of the dialysis, it has shown a good dissolution profile over a period of 16 hours. Then we have titrated the percentage of SLS, surfactant, into the dissolution medium. We used 0.5%, 1%, and 2%. With increase in the SLS, the drug release was also increasing. With 0.5%, it was 57% at the end of 16 minutes, and with 2%, it was showing 83%. But as a part of release dissolution testing, we need at least more than 85, or development purpose, we need more than 85%. So we extended the period to 20 hours. Basically, it was increased to 24 hours. And at the 20 hours, we got more than 95% release. So this is how it was optimized. Next. This is another very good example I have taken from one research article which was published in International Journal of Pharmacy in the year 2014. Here, a very good example, immediate release tablet, meloxicam 7.5 milligram. It was studied by taking 5 generic tablets, and the dissolution profile was compared with the innovator tablet, that is Mobic R, and there are G1, G2, G3, G4, G5. 5 generic tablets were tested using different operational condition to get a discriminating nature. That is what I told you. You can use it for immediate release product also to know a discriminating power of your dissolution method. Okay. So next slide, I will show you how you can see there are difference in the dissolution of G1, G2, and some of them are similar to the RLD sample. Now, which operational parameters has been used to demonstrate the discriminating nature of this test? Madam, next slide. Here, 4 different operational parameters were changed to get the discriminating nature. Okay. Here, first, the effect of cell size. As I explained you, we provide 2 types of cell, 12 mm and 22.6 mm. That is small cell and the large cell. Here, you can see the first graph where RLD was tested using both the cells, and there was no effect of the cell size on the dissolution profile. In the first graph, you can see both using small cell and large cell, the dissolution profile is same. But one of the generic product, when it was tested using the B graph, left side, you can see 2 different dissolution profile were obtained. So this is the first operational condition where larger cell and the small cell was used to get a discrimination in the effect of the cell size on the dissolution profile. RLD has shown no dissolution difference, whereas the generic product has shown the difference in the dissolution. The second operational parameter is the laminar flow and the turbulent flow. Laminar flow means when you put glass bead inside the cell, you will get a laminar flow. And when you do not put glass bead, directly put your tablet in the cell, that time it is a turbulent flow. You can see in that graph, RLD, the A graph, where there is no effect of this laminar flow and the turbulent flow on the dissolution. Whereas the generic product has shown difference in the dissolution profile. So this is the second parameter. Then third parameter, next slide. Yeah, effect of tablet position. Here, free tablet position and embedded. You can see in that picture, this tablet, that pattern A, where tablet was placed on 1 spoon of, that is 6.5 gram of 1 mm glass bead, tablet was placed, and in the pattern D, that cell, entire cell was filled with that glass bead, and tablet was immersed into that or embedded into that glass bead. You can see, obviously, the pattern D will have less dissolution profile, which is shown in that RLD case. And similar experiment was performed with the generic tablet, and you can see the generic tablet here, the embedded position, it has shown 20% drug release. Here, the generic tablet has not shown even a 5% drug release. And here it is showing 60%, that is showing 80% release. So this is the effect that we get. Then effect of open loop and closed loop. RLD was tested using both the open loop and closed loop, and it is showing almost 80% drug release. And even generic product is showing. So which method we have to use? We can see in the next slide summary of this experiment. First, RLD was highly affected by changing the tablet position and slightly affected the open loop and the closed loop setup, and not affected by the cell size and the flow type. Cell size means 12 mm and 22.6, and flow type means turbulent flow and the laminar flow. Whereas the generic flow, product has affected all the FTC, means flow through cell conditions, operational conditions, flow through cell operational conditions. It generic product has almost in all the operational condition except that laminar flow and turbulent flow, it has shown difference in the dissolution profile compared to the generic RLD. The two products behave differently with regard to the cell size. In this respect, it could be concluded that the two products might give different in vivo data. Whereas, based on the open and the closed loop result, it is better to use closed loop to save on time and the chemical used for the dissolution medium. Okay. So this is the summary of this method development that you use before finalizing your dissolution test method. You'll have to optimize the conditions and then you use for your product. So conclusion of this entire experiment is that 3 generic products of meloxicam might not be interchangeable with the innovator product called Mobic. So this was the tested on tablets available in the market, generic, and we could able to see the difference between the generic product and the reference product. Next slide, madam. So this is for the semi-solid product, lotepredinol etabonate ointment, where as per USP General Chapter 1724, three different methods are used. Here you can see the USP type 4 has shown discriminating power also. Next slide, madam. Here it was tested by all the three methods. Method A is with this semi-solid adapter. Method B is for the enhancer cell type 2 apparatus, and the method C is for the France diffusion cell. All this three dissolution medium is giving you dissolution profile and little bit discrimination, but type 4 has more discriminating power. Next slide, please. Madam, next slide. Yeah, here I can explain you how it works. That here discriminating power of that dissolution method using apparatus 4 was tested using, you can see the blue color, red color, and the black color profile. They are of the three different formulations. These three different formulation variant they were given, and they were tested using this, and that is the closed loop, and that is the semi-solid adapter. You can see there is a cup. In that cup you have to put your semi-solid material, that is ointment or cream, and put the filter paper. Here you'll have to do the filter selection, which is the suitable, whether nylon filter paper or glass fiber filter paper or sodium acetate filter paper, which are suitable. That filter paper selection will have to do, and you have to put this adapter inside this. The closed loop, you can see that from the reservoir it goes through the pump, enters from the bottom of the cell, and then from the out of the cell it again goes back to the reservoir. Here the closed loop is changing. What is the conclusion of this experiment is that the compendial in vitro release testing method USP apparatus 4 and apparatus 2 with the enhancer cell are more suitable than the France cell method. This is one. Second, USP4 method is reproducible and more discriminatory than USP2 method. Third, sample loading is superior in USP type 4 and USP type 2 method compared to France diffusion cell method. And USP4 apparatus method showed a strongest logarithmic linear correlation with the rheological properties of that ointment. Okay, next. So this is one example of, I think this is the last example that I would like to show you. This is a PLJ based microspheres where 40 days dissolution has been reduced to 7 days, where the mechanism, how it goes, hydration and drug diffusion and polymer. Here you have to take into consideration the glass transition temperature of that polymer, how it gets, and the polymer erosion happens, and the drug gets released. So next slide, please, madam. Here, it is performed by two methods. One is a real time release profile, and the another is the accelerator release profile. You can see that USP method where it is a closed loop system. Sample loading 10 milligram of the microsphere was loaded on the, and you can see the schematic diagram where red color particles of the microspheres and silver color particle are the glass beads. Flow rate 8 ml per minute, and volume of the dissolution medium 50 ml, just 50 ml. And temperature, two different temperature. Real time means 37 degree, accelerated at 45 degree. And in this real time, you can see it took almost 40 days to complete 100% drug release from the microspheres. And when the same experiment was performed at accelerated temperature 45 degree, it has completed in 6 days. So 40 days has been reduced to 6 days. This is how, but you can see the dissolution medium here. Additionally, 0.0625% of sodium ascorbate were added to prevent the degradation of the drug at accelerated temperature. Okay, next slide, please. And what is the conclusion of the before getting it accepted, they have proved the discriminating nature at accelerated temperature. Here, 3 different formulations were tested using this accelerated condition, and you can see the 3 different profiles were obtained, and conclusion of this experiment that overall the developed accelerated USB apparatus for release testing method has demonstrated the potential of being a fast quality control tool that will assure the product performance as well as assist the product development of the parenteral naltrexone polymeric microspheres. So this is how it works. And this is a small video. Can you run this video? How to prepare the cell? Is it possible? Yes. First, you have to put the rupee bid, then glass bid, then you add your tablet, put the filter paper, appropriate filter paper, close that filter assembly, put it on the cell, and you are done with this. These are the different cell. It is not going further. Stuck. So this is how you have to select your filter paper, put it on this, either use hanger, use this adapter. I think it is not going right. Maybe this. Okay, fine. Go to next slide if it is not working. So finished. Thank you. Thank you very much for patient listening. And if you have any doubts, and you can have that doubt clear on the live demonstration of the apparatus. I will be happy to clear your doubts. And you are lucky enough that ITB University has arranged this program with our Gaia Science people has brought this machine here for the benefit of you. I will tell you, you will not get this opportunity to see this apparatus for live demonstration. Please be attentive and ask whatever questions you have. I will be happy to answer your question. Thank you. Thank you very much.
Thank you so much, Mr. Suhasiwale. Maybe we'll open the Q&A session before the coffee break after the next session first. So our next speaker is Mr. Alexander Hardy, Head of Projects Management at Sotax Pharma Services, Orleans, France. He will present on regulatory applications in the area of drug release using the flow-through apparatus. Please kindly welcome Mr. Alexander Hardy. He has an engineering degree in material chemistry from Polytech Sorbonne, Paris, and PhD degree in sciences of medication and other health products from the University of Strasbourg. He also joined Sotax Pharma Services in 2020 as project leader. Since 2022, he has held the position of Head of Projects Management at Sotax Pharma Services and currently manage the project leaders team and bring technical expertise to support the business development.
Okay. Is it working? Yes. So thank you for this nice introduction, and thanks also to the organizing committee for giving me the opportunity to give this talk today. So can we start the PowerPoint, please? Can we start the presentation? Oh. Okay, thank you. So just before starting the topic of this talk, I have just a few slides to present you about the company I'm representing. So I'm working for the Sotax Group, which is mainly known for the apparatuses we are selling. I mean, we are developing and commercializing. So dissolution testers, automated sample preparation testers, and also physical testing apparatuses. And I am actually representing the pharma services division of Sotax, which is actually an analytical lab which is working as a CRO for mainly for pharmaceutical industries. So we are supporting our customers throughout the whole life cycle of drug product development, from the very early phase with the drug substance characterizations and so on, until the commercial and post-approval phase with routine testing and so on, and through all the different steps of the pure development of the drug products. We have currently 3 labs. The first one is in France, where I am located, and we have also one in India, in Ahmedabad, and one in the US. Oh, sorry. Um, oh, oh, okay. One in the US, close to Boston, in Westboro, which allows us really to have a lab covering, I mean, 3 labs covering the whole world in terms of analytical services. And so we have a US FDA inspected facility. We are registered as a pharmaceutical establishment for both Europe and US. And we are considered as expert advisors for both industries and health agencies in our field. Of course, we have state-of-the-art equipment in our lab, so both from Sotax, of course, for the dissolution testers, and from leading companies for the quantification systems and so on. And also, most of the projects we are leading are related to products for human health. We have also okay. We have also a lot of projects, more and more projects for veterinary products. And so we are able to work with every kind of drug product. Thank you. From very simple ones, such as capsules, tablets, and so on, to more complex ones, such as suspensions, suppositories, semi-solids, medical devices, and so on. And we are giving, I mean, we are supporting our customers through 3 main categories of services: R&D services, GMP services, and support services. So in R&D, as I said, we can do API screening and characterization using dissolution, but also microscopy, for example. Of course, we do solubility studies, method developments, so dissolution method developments, but also quantification method development. We can do in vitro permeation testing for both oral and topical dosage forms, reverse engineering, Q3 characterization, and also we can do IVIVC, so when we want to correlate in vitro and in vivo results, and analytical method automation, both for preparation of samples and of dissolution. And then once the methods are developed, we can also do the validation, transfer of the method. We can do bioequivalency study, of course, stability studies. So we can do both the storage and the testing in course of the stability study of stability samples, and clinical or commercial batch release testing. And actually, all the services we can offer in R&D, we can also offer them as quality control services. And finally, so this is less important in terms of volume, but still it exists. We can also do support services, so consulting, help for troubleshooting and so on, training, audits, and so on. So that was it for the advertisement part. Now I'm going to deep dive into the subject of my topic, which is on the regulatory applications in the area of drug release using the flow-through apparatus, also called USP4. So first, in this presentation, we'll go through slight reminders about the basic of dissolution. Then we will see or RISI because it has already been developed before me, but how we can use in particular USP4 for characterizing API. And then we will see two regulatory case studies on two very different kind of drug products, suspensions and coronary balloon catheter. And after that, of course, we will conclude about all this presentation. So first, an introduction on dissolution. So this is really, again, the basics of dissolution. So because dissolution is often referred to as being kind of a simple analysis, because roughly speaking, you take a tablet, you put it in a dissolution medium in a beaker, you agitate, and you get dissolution. But the truth is that behind that, there is much more science than that, because first, there is not only one way to do dissolution. You have a lot of different apparatuses that exist, and you have a lot of different parameters you can play on to adjust the method. And finally, you can get really valuable information by using dissolution. So that's why it's really important to get interest in it. So actually, this slide is just reminding the basics of bioavailability in general. Because when we develop a method, a dissolution method, when we develop a drug product, it's not for a dissolution apparatus, it's for a patient. So it's always good to go back to the basics and see how it works in vivo. So in vivo, when a patient takes, for example, a tablet, we have several steps. So first, the API will be released from the drug product. Then it will dissolve, and only after that, it will be able to be absorbed through the different membranes of the GI tract. And of course, after that, it goes into the blood, into the tissues, and then you have the action of the API, while already part of it is being distributed and eliminated. And so, when we do a clinical study on a patient, what we do is we will give the patient, for example, a tablet, and then we will take some blood samples at different times from the patient, and we will plot the plasma concentration versus time. So this is what we call actually a PK profile, a pharmacokinetic profile, with this very specific shape. So first we have indeed an increase of the concentration of the API in plasma until reaching a maximum concentration Cmax at Tmax. And then since the API is slowly, more or less slowly degraded, we of course have a decrease of this concentration. And so below the curve here, we have the area under the curve, and we have actually, this corresponds actually to the amount of drug which is absorbed by the body. And so that's what we call bioavailability. And really the important thing to keep in mind here is that this bioavailability is actually governed by the slowest of these three steps. For example, if we have an API which is very poorly permeable, it doesn't matter if it dissolves very fast, because anyway, the limiting step will be the absorption part. On the opposite, if you have an API which dissolves very slowly, of course, the dissolution step here will be the most limiting one. And sometimes we have APIs which are highly soluble, highly permeable, but which release very slowly. And so that's what we call sustained release products. So all that is to say that anyway, when you study a drug product, it's very important to know what is the critical step among these three, and hopefully dissolution can help you defining that. So if I reformulate a little bit here, taking the example of a tablet, what will happen in vivo is that first we'll have a disintegration of the tablet, and this is mainly governed by the cohesive properties of the formulation. Then we'll have the release of the API, which is governed by the type and the proportion of the different excipients you have in your product. And finally, you will have the dissolution of the drug. And this last part here is actually governed by the API characteristics in general. And so the good point is that that's where dissolution can help you while you want to define the API characteristics of your product. So now we're going to see how we can use USP4 to to define the API characteristics. So mainly, unfortunately, this chapter will be a reminder of what was said before me by Dr. Yuda and Mr. Suras, because for characterizing an API, there are two main dissolution techniques. The first one, Dr. Yuda talked about it earlier, is the intrinsic dissolution. So as a reminder, it's a way for you to have an idea of the dissolution of your API while different parameters here are kept constant, such as volume, agitation, and most importantly, the surface. So that since you have a flat surface in this technique, all the physical properties of the API will not have an impact on your results. For example, the size or morphology of your API will not impact the results you will get. So this is essentially for giving you information when you have an API with different solid state properties. For example, if you have polymorphisms, intrinsic dissolution can help you defining whether the different polymorphisms are acting differently in dissolution. And so again, as a reminder, this is the setup that you have. So again, you put API in this part, you press it, you get a flat surface that you put in your dissolution medium. It's agitating, and you get your intrinsic dissolution. And so you obtain, as we saw before, straight lines. And if you compare different polymorphisms, sometimes you can see different straight lines. And by comparing the slopes, you can see you can say whether one polymorphism is dissolving faster or slower or slower than another. So that was it for the intrinsic dissolution. And the second one, which is this time based on USP4, and that was my colleague Mr. Suras explained you just before, is apparent dissolution testing, for which, oppositely to the intrinsic dissolution, here you don't have any modification of the API since it's directly put in the system. So all the physical properties of your API will probably impact the dissolution results you will get. So this is very interesting when, for example, you have an API for which you know that you have different sizes or different morphologies. Apparent dissolution testing will allow you to say if this grade or this grade of API is acting differently. So again, as a reminder, the principle of apparent dissolution testing is here. So again, USP4, so you have your dissolution medium, which is pumped and which goes through this specific cell. As my colleague told you, it's called the powder cell, which is made of different grids and filters, and most importantly of this part where you will directly put your powder. And so, for example, here in this specific case, we compared two grades of paracetamol, and we can see that for the left one, we had a tendency of the API to agglomerate. So for quite a while, it remained undissolved, while on the right sample, it was dissolving very rapidly. So at the same time, you could see that here on the left, you had a lot of remaining unsolubilized API, while on the right, you saw almost no remaining insoluble API in solution. So really, apparent dissolution is very valuable when you want to compare the API properties. And what is really interesting is that you can also easily kind of corroborate the results you have with your API with the ones you get with the final formulation. For example, here, you can test in apparent dissolution testing two grades of API. And with a pretty similar configuration, you can test the corresponding final tablets you get. And so if you have differences on the API, you can say if it impacts or not the dissolution of the corresponding final form. And so, as I said, this apparent dissolution testing can help you see differences if you have different particle size, but also if you have different particle morphologies, such as like much more crystalline particles and amorphous ones. And so, yeah, just to finalize on this first point, apparent dissolution testing in a regulatory environment can be also very valuable in several cases. For example, if you have, again, a tablet and you see that you have more and more out of specifications results when you try to release the batches, and you suspect that maybe a change in the API may be the origin of these differences, then you can use apparent dissolution testing to solve this question and to say whether it's becoming the difference you have on the final formulation come from the API itself or from the final formulation. So that was it for the API characterization part. Now we will see together, as I said, two regulatory case studies where we used USP4. So the first one is on suspensions. So I'm pretty sure you all know what is a suspension, but just as a reminder, it's the fact of having an insoluble active ingredient which is suspended in a proper medium. And so the medium can contain different ingredients, such as suspending agents, viscosity modifiers, surfactants, and so on. And mainly you have two main routes of administration. It can be either orally taken or parenterally. And actually, this specific case is about a parenteral suspension. And so just to go back a little bit in history. So initially, when we first used this solution, it was mainly to test solid drug products. It's only in 2006 that Lilly was the first company to really use dissolution to characterize suspensions. And the next year, in 2007, Sandoz did the same. And actually, today it's becoming more and more common to develop dissolution method for suspensions. And also, the authorities are more and more requesting those dissolution methods to be developed when we want to deposit a new suspension product. So for this case study, the initial point was actually a deficiency letter which was received by our customer after they wanted to deposit a new product, a new suspension. And actually, in this deficiency letter, the US FDA said that actually there was a lack of dissolution data for the test product. And they also tell that USP4 should be the most suitable dissolution testing to develop such a method. But they also tell that maybe USP2 may be worth a try also. And finally, as usual, they were requesting that the method should be optimized for the selection of the medium, the surfactants, the amount of surfactants, and also considering the filters. So this specific suspension was actually containing two APIs. The first one, which was already dissolved in the medium, and the second one, which was suspended in this medium. So what we did is that to really make sure that we were answering all the questions and topics pointed out by the US FDA deficiency letter, we set up a strategy during which we evaluated the different test conditions in USP4 and USP2 also, because we tried both equipments as requested by the FDA. And also we optimized carefully all the selected method. So as a preliminary work, what we did is, of course, we started with a solubility study, which is generally a starting point when you want to develop a dissolution method, because you need to make sure that you have a proper dissolution method to reach the sink conditions and so on. And so we ended up with a phosphate buffer pH 7.2 with some twin as surfactant. And then we carefully selected the filter, and we make sure that this filter was not absorbing the drug, that it was adapted in terms of pore size, and also that there were no leachable coming out of the filter. And so we started, as requested by the FDA, by a USP2 dissolution test using pretty standard parameters, so 500 mL and at 25 RPM. And we got these results. So actually here you have two colors, green and red, because you have both the API which was already dissolved in the medium and the one which was suspended. And so these results were kind of disappointing because, of course, for the green one, which is the active, which is already solubilized in the medium, it's normal that we reach 100% right from the start of the analysis. But for the second one, the red one, which was suspended in the medium, we also reached 100% right from the start of the analysis, which is kind of useless because with this kind of profiles, you cannot use the method. I mean, any kind of batches would give the same results. And this is due to the fact that for suspensions, most of the time USP2 is actually not suitable because it's really highly stressing the product hydrodynamically. So of course you have most of the time 100% dissolved at the start of the analysis. So this test allowed us to put aside USP2 for the dissolution testing of this product, and was a justification to go for the USP4 apparatus. So here again, I will not re-describe, but this is again the functioning of the USP4 testing. And we did again a test in USP4 using again pretty standard the parameters, so 500 ml, 8 ml per minute. And this time we got these results. So again, for the API which was already solubilized in the medium, we had 100% dissolved from the start, which is normal. But contrary to the previous testing, the second API, which was suspended in the medium, this time showed a really progressive profile over 6 hours. So it was way more suitable because with this kind of method, then you can compare different batches, for example. But now the question was, is it really a release profile? In the sense that is the formulation really like included in the profiles we have, or is it only a dissolution profile of the API alone? So what we did is we used apparent dissolution testing to test the corresponding API and to see what results we could get. And this time with this apparent dissolution testing, we obtained these results here. So actually way slower dissolution of the API itself than in the formulation. So that was really good information because first it means that here, here the profile we get are really dependent on the final formulation. And also the second information was that the formulation was pretty robust because let's imagine that we have a change in the API sometimes during the process fabrication or whatever. It is very unlikely that this change in the API will have an impact on the final dissolution of the final product because actually it's the formulation which is governing the release profile and not the API itself. So it was a pretty good information for the customer. So to conclude on this first case study, we've seen that compared to other techniques, so for example, USP2, USP4 can have really a lot of advantages, mainly because it allows to ensure maintaining the formulation integrity during the testing. And thus, the profiles we get are really representative of the release profile of the formulation and not only of the API alone. And so after that, several products were developed by our company and submitted using USP4 technique, including a lot of oncology products. And lately, the US FDA published clear statements requiring that for specific suspensions, USP4 is really the technique to use. For example, for risperidone, naltrexone, azacitidine suspensions, and so on. So really, USP4 is becoming the standard technique to use for testing suspensions. So that's what it for the first case study. Now the second one is on a complete different product, and actually it's on a coronary balloon catheter. Because again, some history. At first, when we started developing drug products, it was mainly simple drug products such as tablets or capsules which were produced. But with time, those products became more and more complex because we had more and more complex diseases to treat. And today we are kind of in a new era of what we call the combination products, which are actually products which have both a mechanical effect and a therapeutical effect because they are containing an API. So here you can see some examples: drug-eluting stents, vaginal rings, coated balloon catheters. Also, some contact lenses can now contain APIs or patches. So these combination products are more and more produced, and also the authorities are more and more challenging towards developing dissolution methods for these new products. So as I said, in this project, we had to work with coronary balloon catheter coated with an API, which was mainly used to treat stenosis, restenosis. So what occurs when we have, for example, an artery that is clogged. And so the principle is that we place the balloon until the place where the artery is clogged. We inflate it. So first we have this mechanical effect. And then we have an API which is deposited at the site of the clogging. And this API is inhibiting the smooth muscle cells proliferation at this place, so that it kind of prevents the reoccurring of a clogging, the restenosis. So this is really an important kind of device because this artery clogging these stenoses are becoming more and more frequent nowadays. And so the starting point of this second case study was again a deficiency letter that was received by our customer from a previous submission of a similar product. And again, so I will not go through all the deficiency letter, but more or less it was the same content as the previous one. So basically, the authorities were asking for a dissolution method to be developed with all the parameters to be demonstrated and justified. And also, most importantly, they requested the discrimination method of the method to be demonstrated. So we used USP4, of course, for this product. And so then we had a specific setup because we needed to maintain the balloons in a specific position throughout the analysis. So we had to to embed the balloons into glass beads here. But anyway, we achieved a success. And we ended up with a method with standard cells, USP buffer pH 7.2 with SDS as surfactant, and again at a standard flow rate of 8 ml per minute. And with the target product, we obtain this kind of profiles, which were very satisfying because they are first very repeatable, so the variability is very low. And secondly, these profiles, you can see that it's like we reach 100% in between 12 and 24 hours. So we can say that the dissolution is sufficiently slow for the method to be expected to be discriminating. So the next step was to demonstrate the discrimination ability of this technique. And so to do so, we had first to define critical quality attributes of the product. So critical quality attributes are actually the attributes which are more likely to impact the quality of the product. So what we said is that since here the API is embedded in a matrix coated onto the balloons, we defined that the ratio between the API and the matrix was a critical quality attribute, a CQA. Also, since the API was available in different states, we also defined that the API state was a CQA. And finally, since several types of catheter were produced on the same sites by our customer, we also defined the catheter type as being a CQA. And this allowed us to select voluntarily produced bad batches that were then used for the discrimination study. And so we used the developed method for this discrimination study, and we obtained those profiles. So here in red, you have the target batch, and all the other batches are bad batches related to the CQA we defined just before. So we had different states of API, different ratios, API matrix, and different type of catheter. So what we can conclude from this slide is that you can easily see that all the other batches are well separated from the target batch, which is an indication that the method is discriminating, and so it's suitable for to being used for QC purposes. And also during development, we had a chance to use this method to analyze a target product, but at end of shelf life. And as you can see, when comparing it with a fresh target batch, we obtain really similar profiles, which means also that the product is pretty stable in time. So that was another good point for our customer. So this method being fully compatible with QC testing, we fully validated it according to ICH guidelines. And then now it is expected to be used for release of batches. So this means that we have to set specifications now related to this method. Since we have a sustained release profile, generally what we have to do is to define 3 different specification time points. The first one here allows you to exclude close dumping. The second one is set to verify the compliance of the shape of the dissolution profile of your batch. And the third one is set to verify the complete dissolution of the API from your product. So for us, in our specific case, we defined these three so the first specification time point was set at 6 minutes, and we said that we needed to have not more than 50% dissolve at this first specification. Then for the second specification, the control of the shape of the profile, we said that we have to have between 50 and 17% and 70% dissolved at 60 minutes. And so for the third time point, at 480 minutes, we say that we have to add at least 80% dissolved to ensure the complete dissolution of the API. And these specifications are actually suitable because if we go back to the different discrimination batches we tested, we can say that if we would have applied these specifications, all the discrimination batches would have been considered as out of specification batches because most of them are not fulfilling the first and the second specification time points. So finally, to conclude on this second case, we've seen that again, USP4 allows to study very complex dosage forms such as combination products. Once again, the interest is that the cell design allows to standardize the positioning of the product throughout the testing. And so it allows you to obtain really progressive dissolution profiles. And even though the products are very complex, thanks to USP4, you can obtain repeatable and reproducible dissolution profiles. And so then, if you are able to demonstrate the discrimination ability of the corresponding method, those USP4 methods can be used for QC purposes for those complex dosage forms. So to conclude, generally on this presentation, we've seen that, of course, dissolution is getting more and more important in the eyes of the authorities, especially for these complex dosage forms. And in particular, the flow-through apparatus, because it's very flexible and also very versatile, is also getting more and more importance for the authorities. Because again, it allows to adapt the operating conditions to the complex properties of these new products. And it can be used both for testing API, for example, for investigation purposes, but also to fully developed methods in regulatory contexts. And last take-home message is that with the increase of the development of complex drug products we see today, one can anticipate that in the future, the flow-through apparatus will become more and more important and more requested by the authorities when you want to develop a new product. So thank you for your attention. If you have any question, I will be happy to answer them. Thank you.
Okay, thank you, Mr. Hardy, for your insightful keynote. And now we will continue to our next sessions, and it will be very interesting as we will witness a live demo of USB Type 4 apparatus. We invite Mr. Simon and team from PT Gaya Science Indonesia, and this session will showcase the practical aspects of the system's operation and application in dissolution testing.
Thank you. Selamat sore Bapak Ibu semua, adik-adik. Terima kasih banyak waktunya masih bertahan di sini. Itu Bapak Ibu udah mulai jenuh karena kita dari jam 8 jam 9 pagi ya. Jadi mohon perkenalkan, saya Simon, juga alumni dari sini. Saya angkatan 94 ya. Jadi di ruangan ini saya merasa terlahir kembali begitu. Ini ruangan ini udah hampir 100 tahun ya, tidak berubah seperti ini. Ya, oke. Pertama saya ucapkan terima kasih kepada Prof. Henny yang sudah mengkoordinasi semua workshop hari ini bekerjasama dengan SPDS ya. Jadi sebenarnya acara workshop ini akan berlanjut ke Bepom minggu depan ya, Selasa dan Rabu. Jadi saya langsung memperkenalkan USB 4. Sebenarnya dissolution system ada 7 ya. 7 aparatus. Yang kita tahu yang paling banyak di dunia adalah USB 1 dan 2. Nah, itu kira-kira ada 90% ke atas lah. Itu pakai USB 1 dan 2. Satu pakai basket, yang kedua pakai pedal ya. Dan ada 3 yang pakai seperti disintegration tester. Ini yang keempat. Ada yang kelima, itu alat yang sama dengan USB 1, 2. Bawahnya dikasih koin ya untuk yang transdermal. Kemudian USB 6 seperti rotating cylinder ditempel biasanya untuk obat turun panas bayi atau apa. Kemudian dissolution rate-nya diukur. Kemudian ada USB 7. Jadi total ada 7 dan ini adalah USB 4 multipurpose dissolution. Kami katakan itu flow through cell ya. Jadi di USB atau IP kita kenal aparatus 4. Kalau di Chinese Pharmacopoeia itu dikenal dengan aparatus 5 dia. Ya. Aparatus 3. Di Jepang aparatus 5. Jadi kira-kira seperti itu. Tapi istilahnya itu tetap sama flow through cell ya. Di sini ada 4 sampai 5 modul. Ini adalah unit utama dissolution-nya dan dikombinasikan dengan pompanya. Nah, ini satu kesatuan yang nggak bisa dipisah. Sedangkan ini adalah auto samplernya yang bisa dibeli terpisah, opsional. Kemudian ini untuk multi position magnetic stirrer, ini untuk closed system ya. Jadi kalau kita running untuk open system fresh media, kita bisa pilih dari media selektor yang ini. Media selektor di sini dia akan memilih media 1, 2, 3 dan media 4 itu menggunakan air untuk cleaning atau pembilasan. Nah, sengaja saya kasih warna untuk membedakan ada 3 media di sini kita anggap pH 1,2 yang merah, kemudian yang kuning pH 4,5 dan pH 6,8 buffer misalnya seperti ini kurang lebih. Dan ini adalah air dan ini untuk menampung waste ya. Kalau kita lihat medium selektor di sini dia akan memilih medium yang berapa. Kalau kita running dengan program dia akan berlangsung secara otomatis ya. Kita coba medium 1. Kamera bisa disorot ke sini ya. Dan Bapak Ibu adik-adik saya akan mempraktikkan cara untuk memasukkan sel-sel ini ke dalam tablet sampel ke dalam sel. Kita keluar dulu. Nah, ini sedang terpasang. Kita akan menunggu jaketnya kosong dulu ya airnya. Setelah kosong kita akan keluarkan ke-7 selnya. Kenapa 7? Karena 6 untuk sampel dan 1 lagi untuk belangko. Ya. Atau placebo kalau misalnya kita pakai kapsul ya. Kita tunggu sesaat. Nah, ke 7 sel ini akan terbuka. Kita tinggal keluarkan. Ya. Setiap sel ini akan kita pasangkan, kita masukkan sampel ya. Saya sudah siapkan sampel tablet, kapsul, powder. Oh, Mr. Suhas ya. Yes, you can help me. Saya akan dibantu oleh Master Suhas. Yes, we prepare the sample now. Oh, gak apa-apa sih.
So this is your cell.
Ah,
This is the
yes.
filter assembly. So this is your
yeah.
cell. You want to show the cell?
Yes.
So
And
this is a
you put
cell.
the ruby bead.
First you put a ruby bead inside. That video also you have seen.
Kita letakkan ruby beadnya. Okay.
See, first the ruby bead. The function of ruby bead is that it will not allow the dissolution medium to go down into the bead. It will allow only dissolution medium to enter from the bottom of the cell inside this.
Ya.
Okay.
Jadi fungsi ruby bead
And
untuk
the ruby bead diameter is 5 mm.
5 mili.
Above that ruby bead, you have to add 1 spoon of this 1 mm glass bead.
Ya.
Okay.
Ini
So,
gelas bead 1 mili.
function of glass bead is then on glass bead, you put one tablet or anything guys, not test a tablet and I'll put
Ya.
now this is the cell and you have to take this filter assembly. There are two parts.
Kita letakkan filternya di dalam. Yes. It's quite hot now.
Can you open this?
Yes. Yeah. Now. You want to put the filter? Yes.
Two parts. This upper part, where you put your finger like this.
Kita letakkan filternya. Ya, yes. Jadi kita letakkan filternya di dalam supaya dia nggak blocking ya. Dan sampelnya siap bisa langsung dianalisa.
This is a lower part of the filter where you
Nah, ini penutup ya bawahnya.
this sieve. And if you are using a control with tablet or a drug product which has some polymers, then some glassful on it so that there will not be a blocking of your filter paper pore. Okay. Yes. Polymers come in contact with the aqueous medium, it swells, and it will block your filter paper. To prevent that blockage, you can put glasses inside this. You can throw it. Put inside. Okay. And then, see, every part of the this thing has a black color pouring. You can see this black color pouring.
Yeah.
Yeah. Kind of black color. So, to get it treated proper, you have to dig into the water or so that get
Yes, we have the water. You can take some water. This all the water, yes, but we need some one cup. Let's have the water. Because it is too dry, I think.
Yeah, either you can apply water on this with the help of your finger, or you can dip this into water and then you put your filter cap on this, like this. Okay, and when you are handling this cell, keep it vertical, don't tilt it. If you tilt it, some glass bead will go below the ruby bead, and in that case, the liquid or the liquid sample that you are testing inside the cell will go into the tube. So when you are preparing cell, you have to remove cell. Cell preparation is the most critical part of the entire differential core apparatus dissolution testing. Okay. And then,
Yeah, we prepare everything ya. Di ada tablet as well. Kita akan coba beberapa jenis sampel. Ya. Ya.
inside the cell, by putting tablet.
Ini kita pakai penjepitnya, tablet holder. Bisa juga kita tanpa tablet holder.
Now he has fixed the position of the tablet inside the cell. Now you can give anything black or green. So you get it here. So that also has to be weighted before you fix it into the cell jacket. Whatever. Okay. So I will dip this and then I'll fix. This is a water jacket. Then I will fix this like this. So I put cell. Then
Yes.
this cap of the jacket. Again, this has a black color ring. So I will wet that with the help of water. Apply some water.
Bisa already.
Then
Ya.
I will put this, and there is a black color ring here also and here also. So I will put the, or I will wet those two rings. I will put this, and with the help of this, I will close it. And when you are preparing this cell,
Kita gitu aja. Oh ya.
or powder. So when you are preparing this cell, put it on the table. Demonstration process and holding. First, you have to fix it properly, like this. Every part has to be fixed properly. And when you are putting the part, you get a fixed part. That shows that you are fixing the cell properly. So this is, and this jacket has this pipe, triple pipe. You have to fix in such a way you have to little bit get this portion also,
Yes.
and to rotate it. There should not be gap between the. Okay, that's good. On. Yes, now it's on. So when you are putting this cell in the cell holder, your pipe should be on the back side and this notch where it will get slide in the cell holder. So when you put this, your pipe is on the back side and then you can push in such a way like that. You can feel a click sound. Okay. So we have put the cell inside the cell holder. Now
Yes.
you want to make another cell?
Yeah, we will try many sample that
This is
tablet and capsule.
capsule.
Yes. And then we also have this tablet with holder.
See this jacket and the cap of the jacket should get fitted properly. Then only it will go inside the cell holder. If there is a gap, then it will not go inside. It is so precise. Even a small 1 mm gap will not allow your cell to go inside. Okay.
Bapak Ibu, ini adalah, hati-hati, ini rubinya jatuh ya. Jadi ini kalau dulu zamannya batu cincin, ini bisa hilang nih. Nah, di sini ada produk kontraseptif implant. Ini real. Ini dimasukkan ke lengan wanita, bisa mencegah kehamilan selama 3 tahun. Ya, ini produknya udah real dan ini dites customer kami. Ini produk punya customer kami. Kita masukkan, kita pasang jaketnya. Jadi ini jaket untuk menjaga temperatur di dalam selnya tetap berada di sekitaran 37 derajat ya. Kita masukkan. Nanti kita bisa lihat bagaimana proses disolusinya untuk semuanya ini. Ini ada tablet. Oke. Dan di sini saya mau tunjukkan juga ada suppository sel ya. Jadi karena saya nggak ada produk suppository, jadi di sini ada satu bagian untuk menghalangi lipidnya masuk ke dalam sel ya. Bisa. Ya, kita masukkan seolah ini adalah sampel suppository. Ini the ya, kita masukkan juga sel suppository dan ini ada sel untuk, can you help me to open?
Yes.
Ini ada sel untuk powder ya. Jadi sel untuk powder, saya semua powder bisa dipakai. Kebetulan saya di rumah nggak ada obat powder, saya taruhnya bubuk kopi ya sebagai sampel. Kita lihat nanti disolusinya. Disolusi kafein.
It's not opening very hard. You have to put water, then only it will become. Yeah. Your powder cell.
Yes.
So this is a powder cell
Yeah.
where you put your powder in that dispersion disk, and then you have to fix.
The last is I would like to apply the cream.
Cream.
Yes.
The semi-solid adapter.
Yes. Jadi untuk krim ini kita akan membuat artificial skin, ya. Nah, menggunakan hole puncher. Tadi kami bikin di luar ya, karena ini di Palu, dan kami dapat artificial skinnya dengan diameter 18 mili ya, seperti ini. Kemudian kita masukkan ke dalam, ya seperti ini. Dan kita sekarang, nah ini untuk penempatan krimnya. Kebetulan saya sini ada krim, ya harusnya ini kita taruh di timbangan, kita timbang dulu, kemudian bobotnya kita catat, dan kita apply krimnya. Di sini ada volume 0,8 ya, 0,8 mili yang sini, dan lebih tepatnya harus ditimbang. Nah, di sini udah krimnya udah harusnya kita isi lebih penuh lagi ya, sampai rata permukaannya. Ini artificial skin biasanya dipakai cellulose acetate.
Cellulose acetate.
Ya.
Or cellulose nitrate.
Ya. Kemudian kita tutup ya, seperti ini. Ada toolsnya di sini.
You can use this.
Ya, ada tools. Jadi kita letakkan krimnya. Ya, kurang lebih seperti ini ya. Ya. Ini ada penguncinya. Buka tutupnya pakai ini. Oke.
Ruby bit. Ruby bit.
Ruby bit inside. Kita letakkan di setiap sel harus ada ruby bit yang warna merah ini. Kemudian kita isi dengan glass bead, dengan spoon ini ya. Glass bead ini 1 cup, jadi udah ada ukurannya. Dan kita bisa taruh seperti ini, ya. Bisa juga tanpa glass bead. It's better not to use glass bead, right?
Yeah,
We
not
use both.
if both. Without glass bead also you can use, without and with both you can use. Okay.
Okay.
By what?
Dan masukkan ke cell holdernya, dan kita letakkan seperti ini. Sekarang kita masukkan ke dalam posisinya, dan kita tutup, dan kita tinggal pencet position close cell. Nah, sekarang kita nyalakan dulu jaket selnya. Dan nanti airnya akan terisi di jaket. Kelihatan mulai terisi. Ya, that's right.
You close, zoom this. Water is getting filled in the jacket. This water from the water bath, which is heated at 38.5 degree,
Ini
will
fluction
enter
collector
in
ya,
this
65 mili
jacket, all the jacket.
setiap
In
tabung ya.
preparation 1, it will take preparation 1 where first jacket water will get filled, then it will go to preparation 2. That time it will say insert the cell. That time you have to insert the cell, and then you have to close, and then the preparation 1, the water will enter into that, and then you start the method. That time dissolution medium will enter inside the cell. Okay, till that time no dissolution medium is entering inside the cell. Okay, so this is how it is happening.
Supaya kelihatan oleh kamera, saya akan mengkolek media ke fraksi yang terakhir supaya di depan kelihatan. Because we have camera on the front, so it is better to.
Okay.
Okay, the cable there.
Open loop system.
Yes. Kamera bisa sorot ke ini, kita begitu kita on, ini disolusi, begitu dia kontak dia udah jalan. Tapi kalau kita pakai metode semuanya berjalan secara otomatis. Jadi
Waiting for temperature is not
yeah,
right.
waiting for temperature.
The dissolution will not start till it reaches the temperature.
Yeah.
37 degree, then only it will start. Now it is showing 32 degree temperature.
Yeah.
So
Because
when
this
it
is for
will
simulation, so we just can start. Dan untuk sistemnya dia akan menarik media. Ini media udah mulai ditarik ya.
channel 1.
Channel 1. Jadi hanya channel 1 yang terbuka. Channel 1 ini akan memompa ya, masuk ke pompa. Dari pompa akan dialirkan ke disolusi. Disolusinya nanti akan dipanaskan 37 derajat melalui spiral stainless ini ya. Jadi ini water bathnya bisa diangkat untuk penggantian air, dicuci segala macam. Di sini ada level sensor kalau airnya kurang. Di sini ada leak indikator, misalnya kalau long term test 24 jam, tabletnya besar, kemudian terjadi clogging di filter, lebih dari 5 bar, nanti tetesan leaknya ini akan keluar. Sehingga operator tahu itu terjadi clogging, seperti itu.
Flow rate 8 ml. Method flow rate 8 ml.
Sorry. Now is pumping, right? Flow rate-nya kita bisa set dari 1,5 mili sampai 35 mili per menit ya. Jadi ini kita set di 8 mili per menit ya. Artinya 1 menit akan terkolek 8 mili. Kalau 10 menit berarti 80 mili. Nah, kalau kita supaya tidak terjadi overflow, tampungannya kan hanya 65. Dia akan mengambil representatif sampel. Caranya dia manajemen waktu. Jadi kalau dalam 10 menit, dia akan ambil setengah menit kolek, setengah menit buang, setengah menit kolek, setengah menit buang, sampai 10 menit. Jadi 10 menit itu benar-benar dapat sampel yang representatif begitu. Ini sampel udah mulai terpapar media ya. Kalau kita deketin mungkin kelihatan.
See that dissolution medium has entered inside the cell. You can see that cell getting
yang nomor 1
dissolution medium, tablet is getting
tablet
disintegration.
Yang nomor 2 itu tablet langsung ke gelas bit. Yang nomor 3 itu kapsul yang isinya juga bits. Yang keempat kapsul saja, kapsul isi powder. Yang kelima itu sampel powder kopi ya. Yang keenam itu krim semi solid. Yang ketujuh itu implan.
Implant.
Ya, kontraseptif implan. Jadi ini karena settingan manual, sehingga di sini semua sampel akan dikolek 100%. Ya, kalau kita
See, you can see that
yes,
excess medium is coming out through this center pipe. For all the cell, the excess medium will go to the west. And in method, you said how much ml to collect? 10 ml?
100%.
1 ml.
Yes.
Okay. So 1 ml it will get collected.
100%. It means that we just manually
Acha, manually.
Yeah.
Okay.
start or stop. Ya. Jadi real USB 4 adalah open system seperti ini. Jadi semua media dikolek ya untuk penghitungan konsentrasi. Jadi kalau kita punya media yang terkolek itu adalah 80 mili, artinya 80 mili kita dapat absorbansi atau pembacaan HPLC-nya dengan volume 80 mili itu. Nanti fraksi yang selanjutnya juga kita hitung lagi sampai fraksi yang terakhir. Jadi konsentrasinya itu diukur dari konsentrasi setiap fraksi, kemudian nanti dikumulatifkan untuk perhitungan total volumenya. Seperti itu. Got some leak.
Splitting not given? Splitting?
No, got some small leak because it's not tightened.
Method you mentioned?
Yeah, because the flow rate is very slow.
Slow.
Jadi kita perlu agak sabar untuk menunggu collection-nya di glass tube ya. Kalau ini di-connect dengan multi-stirrer, dia akan sirkulasi. Perhitungannya di sini sama dengan USB 1.2. Jadi statis volumenya. Sedangkan di sini volumenya tidak terbatas. Selama kita siapkan media, di sini dia akan continuous running terus. Jadi sync condition-nya tercapai. Bisa highlight ke sini. Kameranya. Ke sini apa? Oh ya. Bisa semua. Nah, seperti itu. Oke. Nah, di sini ada clogging ya. Tepatnya di 1, 2, 3, 4, 5. Nah, is clogging happen.
Clogging.
Ya, jadi yang serbuk powder kopinya itu mungkin terlalu jenuh. Saya taruhnya terlalu banyak. Sehingga ini ada clogging di sini.
The filter got clogged. That is why it is showing leakage in this leak indicator test tube. That cell number 1, 2, 3, 4, 5.
Yeah.
Cell number 5 has got clogging in the filter. That is why the dissolution medium is not going out and it is not coming here. Instead of that, the dissolution medium enters inside and it comes back in the test tube.
Yes.
That is the leakage.
Jika Bapak Ibu adik-adik ada pertanyaan bisa langsung ditanyakan ya. Jadi kita bisa langsung tanya jawab. Temperatur udah 36,8 di sini. It's not collect yet.
Method you mentioned.
Because the flow is very slow. The flow rate is 8.
It is not sitting on this. It should come. This is the row.
Oh, yes. It's still. Split to west.
Split. Yeah, to. Now it is started.
Okay.
See, it is now dropping.
Yeah. Okay.
See, now that's it is getting collected in the test tube. You can see the drop that is going.
Ya. Tadi settingannya ke west semua. Jadi dia nggak collect, sekarang baru berkolek ya. Dan yang kelima karena itu yang clogging tadi dia nggak keluar. Kalau terjadi clogging ini harus di stop karena ini hanya simulasi, kita lihat hasilnya saja. Itu karena tabletnya berbagai jenis sehingga warna sampelnya juga bervariasi ya.
Getting
yes.
This one.
Number 5.
Number 5 is not getting collected because it is clogged.
Yeah.
The cell is clogged because the filter paper has clogged because of some excipient, that is polymer, and it has clogged the filter paper. That is why flow is not coming in the 5 number cell. You can see here. This rest of the cell, it is dropping. And it is getting collected here in the dissolution medium is collected in the cell. And one thing that you have to see to it that the waste you are using, the tube should be kept on the upper side. It should not dip in the waste solution. Otherwise, it will create a back pressure and it will again disturb your collection. Okay. Now it is flowing at 8 ml per minute. If your sample time point is 10 minutes, it will collect 80 ml. And if you are given, you want to collect 10 ml inside the test tube, it will collect 10 ml. 70 ml will go to the waste from each cell. Okay. The split ratio will automatically change. How much ml you want to collect, accordingly it will split the ratio.
I change to media number 2. Jadi barusan saya ubah lagi, sekarang medianya di nomor 2.
And this pipe is long, then you can put it at a distance like this. So that this will it should be like this. It should not be dipping inside the solution. Otherwise, it will create a back pressure. Okay?
Ya. Ini kalau kita running dengan program, ini berjalan secara otomatis ya. Karena ini hanya untuk demo show, kita mau memastikan bahwa medium selektornya ini bekerja dengan benar. Ya. Jadi misalnya kita bikin program jam pertama pakai medium 1, ya, sama dengan gastric acid. Yang kedua dengan pH 4,5. Kemudian yang ketiga dengan buffer fosfat.
I will explain closed loop.
Yeah. Which one?
I will explain them how closed loop will work.
Oh yes, you can do it.
So this is a closed loop system where this is a reservoir. You can see there are 3 tubes. You can see 3 tubes are there. Can you focus?
Yeah.
Yeah. There are 3 tubes. One tube is for the inlet reservoir The dissolution medium is going to the cell. Another tube is for that dissolution medium. After dissolution of the product inside the cell, it will come into the second tube. And the third tube is for taking out the sample from this reservoir. The difference between the USB type 4 and the closed loop system of your apparatus 1 and 2 is that in apparatus 1, sampling is done from the dissolution vessel. Here, the sampling is done from this reservoir. It is not from the cell.
Yeah.
That is why it will not disturb your hydrodynamics. In apparatus 1 and 2, it disturbs your hydrodynamic. What happens during the dissolution, the cannula will go inside the vessel and it will take the sample. So that time the hydrodynamic will be disturbed. Whereas my dissolution is happening inside the cell. Okay, dissolution is happening inside the cell and my sampling is happening from the reservoir. And I have put that magnetic needle bar so that this is a multi-point magnetic stirrer where all the 7 cells will come here, dissolution medium will come here, and sampling will here in this fraction collector. I will replace this splitter with a series module. Okay, and it will collect directly. This rack will be replaced with the HPLC rack, a smaller HPLC vial. You can collect 1 ml directly into the vial. So this is how it will work, closed loop system. At this moment, this is open loop system is there. Closed loop system, how it will work that. So in closed loop system, whatever this inlet tubings we have collected to this multimedia, that is a media selector, you will connect to this bottle. Inlet and this outlet, you will connect to the bottle. Okay, and you will remove this and there will be a series module. In that picture, you might have seen that in the slide. So this is how the closed loop system will work.
Okay. Dan aplikasi lain flow through cell ini tidak terbatas untuk farmasi. Jadi kemarin saya juga mendemokan ke pabrik rokok, ya, di mana untuk terapi orang yang melepas rokok itu bisa pakai tobacco pouch, ya, kurang lebih seperti tea bag seperti ini, ya. Jadi untuk aplikasinya mereka mau cek kadar nikotin yang ada di smokeless tobacco pouch ini. Tobacco pouch itu mereka letakkan di bawah lidah. Jadi kayak diletakkan, mereka menghisap sari tembakonya seperti itu, ya, karena mereka suka dengan katanya kalau orang melepas rokok itu kan lidahnya pahit atau apa. Nah, kurang lebih seperti itu mereka bisa pakai ini untuk terapi. Kemudian kita letakkan tobacco pouch ini di dalam dan kita timpa dengan glass bead lagi dan kita masukkan filter cupnya dan kita masuk ke sel. Seperti itu.
You will have to use that artificial saliva medium, pH 7.4.
Iya. Dan mereka
You
menggunakan
can
media.
prepare that medium and you can see how the drug is getting released. The nicotine is getting released in your mouth,
Ya.
in saliva. Same way it will mimic that. Correct?
Artificial saliva itu ada formulanya,
You can
ada
click
7 macam bahan
there.
untuk
You can
membuat
click
artificial
there.
saliva. Ya, kemarin kami lakukan, ya.
So any question? Open loop, closed loop? For calculation purpose, again, closed loop, it will be same like your type 1, type 2 apparatus. But for open loop, each time point, how much dissolution medium that you are passing, that much volume will have to do the calculation. For example, for your first time point, your flow rate is 10 ml per minute. First time point it is say 10 minutes. So you are passing 100 ml. 10 ml flow rate multiplied by time point. That is 10 ml for 10 minutes. So you will consider 100 ml volume for your calculation purpose. Got it? And suppose second time point is 30 minutes. Okay. So in that time point, so first time point is 10 minutes, second time point is 30 minutes. The difference is 20 minutes. So in 20 minutes, at the flow rate of 10 ml, you will pass 200 ml. Correct? 10 ml multiplied by this 20-minute difference. So for second time point, you will consider 200 ml for the calculation of that dissolution. That is how you will do the calculation of your open loop system. And in open loop system, in closed loop system, you calculate dissolution profile by cumulative method. Okay, every time point you will add cumulative 10 minute, 20 minute, 30 minute, like that. Here also, in open loop system, you can do the calculation by 10 minute, 20 minute, 30 minute, like that, by adding what is the drug release in first time point, that is 10 minute. You add what is drug release in 10 minutes to the second time point. So 20. So cumulative, you will get a graph like closed loop. But if you want to see how that differential, it reaches to the maximum and then it goes down. So direct release, whatever you get, you will get that differential curve. So there is two columns. One is the direct drug release, and the second column in the Excel sheet is the cumulative drug release. So if you want to see the differential curve, you can get that graph of differential curve from the first column, where direct release you are calculating. So this is how it works. Correct? Getting it? Any question? You want to show anything?
Yes.
Yeah. This is the temperature.
Ini adalah temperatur calibration probe. Jadi kita ganti probe yang di atas dengan ini dan kita tancapkan temperatur measuring probe-nya di lubang ini untuk mengecek aktual temperatur yang ada di dalam media, di dalam selnya ini. Ya. Jadi kita letakkan di sel yang di tengah seperti ini. Pada saat kita kualifikasi mengecek temperaturnya. Jadi untuk alat ini kita melakukan kalibrasi flow rate untuk pompa, dan untuk di unitnya kita hanya cukup mengkalibrasi temperatur yang ada di media. Mungkin Bapak Ibu tahu kalau di USB 1.2 pedal ada prednison tablet dari USB untuk PQ-nya atau DPVS ya. Dissolution Performance Verification Test. Kalau di USB 4 ini tidak perlu ya.
So apparatus 4, you do not need to go for chemical calibration. Only flow rate and temperature.
Yes,
only two parameters you have to do the calibration. Flow rate and temperature. Temperature of the cell, dissolution medium that is going. And the flow rate, whether 8 ml flow rate or 16 ml flow rate, as per that flow rate, you have to measure, you have to collect that dissolution medium passing through your cell in bypass position. And if you have set 8 ml, in 1 minute it should drop 8 ml. If it is 16 ml, in 1 minute it should drop 16 ml. That is the flow rate calibration.
Dan untuk yang clogging tadi kan Bapak Ibu lihat di sini clogging ya. Nah, dia nggak akan luber ke luar karena di sini ada drainnya. Ada drain kecil, mungkin kalau bisa disorot, kalau saya buka seperti ini. Nah, ini drainnya akan masuk ke lubang yang di sini, dialirkan ke belakang ya, ada drainnya. Jadi nggak perlu khawatir akan luber ke luar, karena ini dia masuk ke dalam, dibuang ke drain tube seperti itu. Dan di belakang ada drain, nggak kesorot ya. Dan ini sistem udah kita matikan karena udah penuh ya. Jadi misalnya kita kolek di sini 60 mili, 60 mili berarti misalnya 8 kali 4 8 kali 5 misalnya 8 mili per menit kali 5 menit itu berarti ada 40 mili. Nah, kita ukur konsentrasinya di sini konsentrasi 40 mili ya. Nanti kita kumulatifkan dengan fraksi-fraksi yang berikutnya fraksi 5 menit, 10, 15, 30, 60, seterusnya sampai kita dapat nantinya konsentrasi di mana kita kira-kira rilisnya sudah apakah 80% seperti itu. Nah, kalau misalnya kita gabungkan semua fraksi ini konsentrasinya rilisnya belum 80%, berarti kita masih harus tambahkan durasi tesnya. Kurang lebih seperti itu. Jadi di sini ada proses method development, ada kita try and error ya untuk mencari durasi tes untuk produk yang baru.
Here, told them about this color coding.
Yeah.
Color coding of each.
Oh yeah, you can.
See, in this apparatus, you can see there are a lot of tubings. You know, there are a lot of tubing going inlet, going outlet, going to the reservoir. So you need not to get worry about that because each cell and each tubing has a color coded end fittings like this. See, brown color, red color,
Yes.
see, this is a first is a brown color, then it is a red color, third one is orange, fourth one is yellow, fifth one is green, sixth one is blue, and seventh one is pink color. So right from the inlet of the reservoir, these colors are there. One number goes to the one number inlet, one number of the outlet of the pump goes to the one number inlet of the cell, and from this again one number outlet. So there is a complete traceability. There is a complete traceability that color coding of that one. You have to collect one number, connect one number to the this media splitter.
Kita simulasikan close system.
Sorry, this media selector, then goes to the inside, coming out, going out. Okay, so there is a complete traceability, entire tubing. You need not to worry on that. But while putting all the cell or all the connections while using this closed loop also, you have to see that you are connecting inlet of one number to the inlet outlet of the one number in the bottle also. Okay, so when you are connecting inlet outlet, connect that. And even when sampling is done, you have to connect that sampling port to the syringe. There is also color coding, is same color coding is there. So entire system has complete traceability, which cell is working and which sample is getting dissolved. Okay.
We are preparing for the closed loop system.
Okay, you are preparing.
So that the audience easy to understand.
Easy to understand.
How is
correct. And there are these filters also that you can put it directly there.
Yes,
inside.
directly. And we will remove the
You want to
fraction
remove this?
collector. Yes.
I'll remove this.
I think no need.
Stop.
Because we already stopped it. Now
stop.
we will
Remove this.
yes.
So now we are converting open loop system into a closed loop system. So he will, first thing that you will have to remove this splitter so that your all these tubings will go into the reservoir bottle. So you have to put this bottles like this.
Nanti ini geser. Ini geser. Ini geser. Geser aja. Ini tukar ya. Ini geser, angkat. Angkat.
So you have to put in between.
Ini angkat. Geser.
To
This
put
one
that.
we remove.
So when you are using closed loop system, this multi-point magnetic stirrer with your reservoir bottle will be beside the pump. To connect all this, will connect.
Yes, we connect it.
First, we'll put the bottle there.
Rizky, ini aja lepas.
Give the bottle. We'll connect there only. Bottles. Give the bottle. So we'll connect. Will they show that? Yeah. We'll put the bottle first here.
Yeah. Fill the water. Buka dulu botolnya. Buka semua.
Let them show the camera, na?
Yeah.
How they are connecting. So there are 3 tubings. You can see for each reservoir there are 3 tubings. 1, 2, and 3. One for the inlet of the dissolution medium. Another is for the outlet coming back to the reservoir. And third one is for the sampling from the reservoir. We're going to put this series model also? No. Series model? No.
Only
Only
closed system.
this
Only closed system.
process.
Ya tutup lah ya, Rizky. Yang udah. Jadi sekarang kita akan simulasikan bagaimana workflow-nya untuk closed system. Kita isi dulu media, misalnya di sini ada 250 ml ya. Ya. Jadi semuanya harus seragam. Jadi semua media ini yang akan disirkulasikan di dalam sel. Satu. Ini yang sini ya. Kita pasang sesuai warna. Jadi ada 3 hole, 1 inlet buat media ke pompa, 1 untuk kembali dari disolusi, 1 lagi buat sampling. Jadi untuk close loop system ini kita dengan konfigurasi ini pakai yang manual sampling ya, pakai siring. Ada opsional untuk auto samplingnya juga.
Inlet here is not connected.
On one.
You have that tray also. Expensive oil racks. Sampling rack here and all that.
Misalnya kita anggap sampel yang sama, kita baru start, kita tinggal nyalakan pump on. Kemudian di sini sampel akan langsung disirkulasi ya. Medianya tersirkulasi dari sel ya. Jadi media selektor ini nggak akan dipakai lagi di sistem ini karena media selektor hanya untuk yang open loop system. Untuk closed loop system, workflow-nya seperti ini ya, Bapak Ibu, adik-adik. Jadi media akan ditarik dari botol, kemudian masuk ke pompa. Dari pompa akan diteruskan ke disolusi. Kemudian dari disolusi sel akan kembali lagi ke botol. Ya. Jadi ini closed loop system seperti ini runningnya. Nah, pada saat udah running dari titik nol tadi, pada saat pompanya on, kita bisa lakukan sampling. Bisa pakai shearing. Misalnya untuk fraksi 5 menit, kita bisa langsung tarik ya. Mungkin kelihatan ya. Nah, seperti ini. Kita bisa ambil misalnya 5 mili atau 10 mili. Kalau misalnya kita perlu ada yang pakai media replacement dengan volume media yang sama, kita kembalikan ke botolnya seperti itu. Demikian pula untuk nanti fraksi ke 10 menit, 15, 30, 1 jam atau dan seterusnya 2 jam misalnya. Oh iya. Untuk closed loop system konfigurasi ini manual sampling. Tapi ada satu device buat auto samplingnya, opsional, ada. Di sini ada hot plate. Kita biasanya setting di 40 derajat supaya untuk menjaga suhu di media sekitar 37. Kemudian juga dilengkapi dengan magnetic stirrer yang kecil. Oh, we don't put it.
You need not to maintain the temperature of these bottles, because once that dissolution medium enters into the cell, automatically it will take 37 degree. Because this is exposed to the atmospheric temperature, it may be 40 degree, 37 degree, no matter. So before entering into the cell, it will attain 37 degree.
Ada pertanyaan lagi, Bapak Ibu, adik-adik?
So here in this closed loop system, there is no drug going out. There is, because we have disconnected this. It is like now apparatus 1 and 2. It is getting circulated inside only. No drug will go out in like open loop system. Okay. Your drug reservoir goes to the inlet of the pump. From outlet of the pump, it goes to the inlet of the cell, and from outlet of the cell again coming into the bottle, reservoir bottle. So it is a closed loop circuit complete.
Yeah.
Clear?
Oke, Bapak Ibu, adik-adik.
Any question? Anyone want to know?
Kalau enggak, kami kembali kepada MC. Anything else you want to show? Yeah, I think it's enough. Sistem ini sudah ada 10 sistem di Indonesia ya. Jadi beberapa customer kami juga hadir di sini. Terima kasih atas supportnya. Ya, kami sampaikan di sini terima kasih banyak atas perhatiannya dan untuk workshop kali ini. Jadi selain kita belajar juga semoga menambah ilmu ya. Ada bahasa Inggris dari India, ada bahasa Inggris dari Prancis. Oke, terima kasih semuanya.
Thank you, Mr. Simon and his team from PT Gaya Science Indonesia. And this session will end up our
Thank you.
conferences today. And ladies and gentlemen, this brings us to the close of today's Dissolution Science and Applications conference workshop. On behalf of the organizing committee, I extend our heartfelt thanks to all speakers, participants, and partners for making this event a success. We hope you found today's session insightful and valuable. Wishing you all a pleasant evening and continued success in your professional endeavors.
For your appreciation, please scan the barcode that will be shown in the screen so you can get a certificate for this event. And before you go home, you can take a snack behind your seat. And if you want to try to see the instrument more closely, you can go to the front and try the apparatus. Thank you and see you at Disso Asia 2025.